dignitaries to kindly grace the dais. Our Honorable Vice Chancellor, Lieutenant General Madhavi Kanetkar ma'am, to please grace the dais. Honorable Co-Vice Chancellor, Maharashtra University of Health Sciences, Dr. Milind Mukund, sir. Our co-host for the event, Director, National Center for Cell Sciences, Dr. Mohan Vani, sir. And our keynote speaker for the day, Dr. Mitali Mukherjee, ma'am. I request you to kindly grace the dais. Our second speaker for the day, Dr. Supriya Bhalerao, ma'am, would be joining us shortly. I welcome all the dignitaries right here, wholeheartedly from the Maharashtra University of Health Sciences, Nashik, in collaboration with National Center for Cell Sciences, Pune. A round of applause for all the dignitaries who have made here in spite of the Pune traffic on time. I also warmly welcome our kickpins from MUHS, Brigadier Subodh Malgun, sir, and Dean, sir, MPGI. We welcome you here, sir, for the event. And I see happy faces here of the teachers, the PG, PhD students, residents, who have been nominated by their principals and well curated to the top 50 to be seated in this auditorium. I warmly welcome all the august audience here for this workshop. A clap for you all too. They have made it to the top 50 months. So today, the workshop theme, as you all know, is Integrative Health and Personalized Medicine through Ayurgenomics. I do understand all of you here are curious to understand and unravel the depths of integrative medicine. And we all have our doubts, our understanding, and the curiosity. So, to pay a tribute to the ritual of lamp lighting, I would quote Brihadaranyaka Upanishad here. Asatama Satgamir, Tamasoma Jyotirgamir. From ignorance, lead us to the truth. From death, lead us to life. So, this auspicious lamp lighting will clear all our doubts, I suppose, and I now welcome the dignitaries to kindly lamp the light. Yes. Yes. Request the dignitaries to kindly take their seats. Being the NCCS, we were strictly told that the lamp lighting be a very brief event and that we focus on the science part of it. So that then 137 we would have at MUHS now. Yeah. Okay. Thank you. Our keynote speakers have been here all the way from Jodhpur, and it is an honor to welcome them on behalf of the Maharashtra University of Health Sciences and the National Center for Cell Sciences, Pune. I now request our Honorable Vice Chancellor, Lieutenant General Madhavi Kanetkar ma'am, to kindly felicitate our keynote speaker for the day, Dr. Mitali Mukherjee ma'am. second speaker would be joining us shortly. She'll be felicitated when Madam arrives. Okay, now moving into the crux of it, this entire complex set of words which intrigues everyone, integrative health and personalized medicine through Ayurgenomics. This will be simplified for the teachers and students and research coordinators of MUHs, because someone there at the top realized that 10 years down the lane, it's going to be integrative health, and that someone is none other than our dynamic and supremo of all, Lieutenant General Madhuri Kanitkar ma'am, who was behind us do something integratively. Since 3 months, madam has been following up the program, the speakers, and the breakout session, the theme. I mean, this is the outcome of, as we say in Marathi, the Vichar Manthan. You know, the final outcome comes out of a lot of brainstorming. So this is the brainchild of our beloved ma'am. Thank you, ma'am, for giving us this opportunity. I would now request our head of the University Research Department, Brigadier Saurabh Sen sir, to kindly give a brief background and introduction of the workshop.
So good morning all. I'll keep it brief. Like the previous compare has just announced and informed all of you, the plan is that this is the place, this is the event which is going to be the melting point. And I have been also learning what in the modern medicine we all learned about phenotypes and genotypes, and what you have in Ayurveda, the prakritis, Vat, Pitta, Kapha. They are existing, they were always there, this knowledge was there, let it all integrate. And that is what Dr. Mitali Mukherjee told us, that let it be something integrated as far as, you know, so as to bring out something personalized for each person. So the aim is here to listen to the speakers. We have such a wonderful, get together where we have experienced speakers in the fields of basic science, modern medicine, Ayurveda, and we have participants from various health science colleges. We'll all first listen to the experts. We will have some brief, presentations followed thereafter by some of the experts in their fields, like we have from, diabetes, on oncology, on Ayurveda, and also on genetic disorders. And thereafter, we will get into various breakout groups. There'll be brainstorming. We will expect all of you to participate, understand, contribute, so as to come out with some broad guidelines and some deliverables at the end of the talk. And aim is to carry forward. This is not a kind of an event which ends today. This is the beginning, and let's carry it forward. Thank you. And now I'll request the dignitaries to come down to the audience. And only I'll request Dr. Milind Nekam to come over and deliver the next talk on the insights in Ayurveda. Our Pro Vice Chancellor, please.
Thank you, Spencer. First of all, I will thanks to our Vice Chancellor who has arranged the first integrative workshop on the personalized medicine and Ayurgenomics. Charak has already described the theory. Yasya yasya hi anga avayavasya bija bhaga upatapto bhavati tasya tasya hi anga avayavasya vyadhir or vikruti upajayate. So on the basis of this, we know that the Charak has already described bija, that is spermatozoon and ovum, bija bhaga, that is chromosomes, and bija bhaga avayava, that is the genes. So to elaborate all this with the modern medicine, the modern science has developed its own path, and we are going to collaborate to learn how the Charak has invented that, and the theories are to be implemented in this. Secondly, the Prakriti. सगळ्यांना माहिती आहे प्रकृती ही उत्पत्ती होत असताना शुक्र शोणीत संयोग ज्यावेळेस होत असतो त्याच वेळेस प्रकृतीची उत्पत्ती होत असते. ही प्रकृती 7 प्रकारची ग्रॉस मध्ये सांगितलेली आहे, ज्याच्यामध्ये वातज, पित्तज, कफज, त्याच्यानंतर वात पित्तज, कफवातज आणि वात पित्तज, कफ पित्तज आणि त्रिदोषात्मक अशी प्रकृतीचं वर्णन आलेलं आहे. तर प्रकृती ठरत असताना आपल्याला आयुर्वेदात आपण बघितलेलं आहे, दुष्यम देशम बलम कालम अनलम प्रकृती वयः सत्वम सात्वम तथा आहारम अवस्थाचे पृथक्विदा. ह्या सर्व गोष्टींचा विचार करून प्रकृती ठरत असते आणि प्रकृती ठरत असताना त्यावेळेस त्याच्यामध्ये जो काही त्याचा पॅटर्न आहे तो ठरला जातो आणि त्याच्यानुसार उत्पत्ती होत असते. त्याचबरोबर ज्यावेळेस आपल्याला हा जो विचार करायचा इंटिग्रेटेड हेल्थ बाबत, तर याच्यामध्ये आपल्याला एक दोन विचारधारा आपल्याला करणं गरजेचं आहे. आपल्या याच्यामध्ये शास्त्रामध्ये सांगितलेलं आहे तर बीज शुद्धी आणि आहार विहार या माध्यमातून जर आपण त्या बीजाला जर प्रॉपरली आपण त्याला नरिश करून प्रॉपर जर त्याला आणलं आणि त्याच्यामधून जे संतान उत्पत्ती होईल त्याच्यामध्ये आपण दोष हे आपण काढू शकतो. आणि आज आपण तेच इथे पाहणार आहोत. तर मी सर्वांना माझं अभिनंदन करतो, या ठिकाणी वर्कशॉप अरेंज केला. धन्यवाद मॅडम. थँक्यू.
Thank you, sir, for those wonderful insights. The very existence of our university stems from interdisciplinary roots. Our Pro-Vice Chancellor is from an Ayurveda background, an expert and thing in Ayurveda, and our head of the department is a microbiologist, and of course, our madam being the pediatric nephrologist. So, the existence of our university basically stems from a very interdisciplinary melting pot roots. With that in mind, I now request our Honorable Vice Chancellor Madam, Lieutenant General Madhuri Kanikar ma'am, on the dais to kindly express her welcome address for the day. I welcome you all with a round of applause, ma'am.
Thank you, Shweta. My Vice Chancellor, Dr. Milind Nikum, Madam Godbole, Dr. Vani, Madam Mande, Mitali, and the Dean of URD department, faculty of URD department, but most important, all of you who have made it to the thing. I see my chair professor Anuradha here, my expert Dr. Leena Gore. I mean, you just see what a melting pot we have created out here. And that is the first step to integration. Each of us come from a very diverse field, but we are trying to all align our energies to one goal, and that is health and wellness. So if you have to do that, we need to have a very, very integrated approach. And my own journey, because majority of you are from both Ayurveda and modern medicine, I thought I must tell you why we sort of started thinking on this. It all started with my first exposure to Dr. Mitali when she was with IGIB, and we were working on finding solutions in this, and one of my own students was doing a PhD with IGIB, Ranjit Khairgaonkar, for looking at something which is very unrelated, Kakut. But while doing that, I had a chance encounter with her, and for the first time, she told me how you can approach medicine from a different perspective of what is ancient and known knowledge. So should we now go in only one direction, that we have a disease and then we find the genetic markers and then we go into treatment? That's one way. But we know, and with more and more knowledge coming in, we are aware that it's not so simplistic, and that there are multiple factors which affect your final wellness and illness concept. The same old host environment agent triad, which we've always concentrated on the agent, how to treat disease through the agent. But the host characteristics, the environment, what your surroundings, have not been given adequate importance in modern medicine. And I thought this was one way that can we actually leapfrog to find solutions for the world by looking at ancient wisdom, and that is how I started reading myself about the Prakriti or the doshas, trying to understand. I'm nowhere near understanding, and I'm just a student, as most of you would be. But that is what it is. We are lifelong learners. So let us look at the problem which we've not been able to solve for so many years from a different perspective. And that was why this was born. And I went into the university thinking, how can I bring in integrated? And I'm extremely lucky that my Pro-Vice Chancellor is an Ayurved, he was a dean of an Ayurved college, and he brings a very different set. And most of the time, we are brainstorming and learning from each other. So this is actually the genesis of it. Then we threw in into the melting pot data science people, we threw in scientists, we put in more Ayurved. Shweta here is an Ayurved and she's working on integrated research. Dr. Sen, who's my dean, like she said, is a microbiologist and a pure science person. So we are putting everything to look at problems in a very different way. There is Anuradha, who's joined us as a chair professor, who's been working on science and genomics for cancer. But we also know, like I said, that we have a very different traditional knowledge, which probably they did not know it as genetics, but they probably had an observational value to it. And at many years ago, Mitali, we were looking at seeing whether we can predict high altitude problems and whether there are any signatures available for acclimatization using the Prakriti. So this was something which I think we were a little ahead of our times and we couldn't push it through. But having come here, I feel, and you people with a huge resource, I thought we should put this workshop together. And I'm extremely grateful to Dr. Mitali to mentor it, and Shweta, my Pro VC, Dr. Avale, and everyone to really put it together, and for the regional center Pune to co-host it. Because we said if you're doing something new, we must have the majority of the people coming with a thought process in a larger city which has Ayurved and medical people from different faculties. And that is why we chose Pune. And I'm extremely grateful to NCCS for giving us the venue to use, because we have an MOU. But unless we keep talking to each other, we're not going to make headways. And I'm extremely happy that Dr. Mrs. Mande is here, who's going to sort of, bring in a different perspective to our whole issues together. So in the end, I'd say this is a workshop. I mean, you're going to work, but you're going to shop. And when you go for shopping, you go with lots of empty bags, and you're not going to feel happy if you come back with an empty bag. You have to have struck a good bargain, right? That is the pleasure of coming back. So I don't want this workshop to be a CME, which is come, meet, eat, and then go back and sleep over it. So I want you all to be jumping into it, that I'm going to take back something in my bag and I'm going to, give my best to it, to come out with projects which we can collaborate together. And I assure you, the university is waiting for you all to put up such projects, and we will give you the seed funding, give you hand-holding to write up those proposals and take them forward to find answers to old problems, new solutions. Thank you so much. All the best. Jai Hind.
मेरा वचन ही अनुशासन है. So you got it. This is not a workshop. Thank you for making it so simple for us now. Because yes, we do have sessions. We do have hand holding up to a certain limit. Then we leave you all to breakout rooms. Dr. Sen sir would be explaining it post lunch. But be attentive, because we're going to actually make you work at the workshop. Okay, now I request, before that, we've been joined by Dr. Supriya Balera, ma'am. Thank you for making it, ma'am. He is our second lead speaker for the day. We will have the session after the keynote discuss. Thank you, ma'am. And now I invite Dr. Manasi to introduce our keynote speaker for the day, Dr. Mitali Mukherjee.
Good morning. It's indeed an honor to introduce Mitali ma'am. Mitali ma'am is a professor of Department of Bioscience and Bioengineering at IIT Jodhpur. She is the former chief scientist at the CSIR Institute of Genomics and Integrative Biology. She has been the convener of the Indian Genome Variation Consortium project, which provided the first comprehensive genetic landscape for the Indian population. She has been involved in developing a new area of research, Ayurgenomics. That's why she's here to kind of help us promote it, and which aims to integrate Ayurveda with modern medicine through genomics. She was the program director of CSIR Ayurgenomic Unit Trisutra. Presently, she is involved in developing Center of Excellence in Integrative Precision Health, wherein the aim is to develop a data science-driven framework for population and individual risk stratification and early actionable precision health interventions. One of the sub-projects in this initiative is also Ayurtech that combines electronics, digital health, and AI and multi-omics approaches for realizing evidence-based Ayurveda solutions in the in a transdisciplinary framework for implementation of integrative medicine in different technical settings. Mitali ma'am is also coordinating a project in the Jodhpur City Knowledge Innovation Cluster called the Thar Designs, Desert Ecosystem Innovation Guided by Nature and Selection. The aim is to carry out an integrated study of the desert ecosystem to propel discoveries, inventions, and innovations for addressing health challenges in the arid regions. So without further ado, I will request Mitali ma'am to please come on stage. And thank you.
Good morning. Yeah, am I audible? So just you have to excuse me a little bit of a throat problem, continuous travel, but it's really a pleasure to be here and to be able to interact with you and talk about the things which interest me and have been passionate for over 2 decades. And I would really like to thank Dr. Madhuri Kalitkar, Lieutenant General Madhuri Kalitkar, for giving me an opportunity, because we've been interacting for many years in various ways, but not really trying to, we've not been able to make headway into working together. But all these things really take time when we are in different settings and all. And Dr. Mohan Wani, I met him last week and I told him it was a coincidence. I met him and I said, I'm staying at your guest house, and Dr. Mohan Wani and Dr. Shamila, we've all known each other for long years, Dr. Papat and Supriya. So it's really, and I've been in Pune for many years off and on, because we were registered as guides from Pune University, and I've worked with Sanjay for over 10 years in Vadu. So it's a pleasure to be here. What I'm, I think the left side will be gone.
It's just this
The first slide, I hope. So this is our campus, and all of you are welcome whenever you come to Jodhpur. Please visit us. It's 884-acre isolated place, a little away from the city of Jodhpur, with lots of wildlife and all, and quite a happening ecosystem now. So I actually took a big risk to leave a very comfortable 23 years of existence in IGIB and just shifted there. Dr. Mande was there. She reviewed our department recently. So in IT, I kind of was invited to develop a transdisciplinary center of excellence in integrative precision health, primarily because, as ma'am mentioned, data is the key for making discoveries, and data along with artificial intelligence can scale up and globalize a large number of things. But then we need to have a platform where we could have lots of disciplines able to cross-talk with each other. So IIT Jodhpur has developed a school of artificial intelligence and data science, where I am a faculty of bioscience and bioengineering, but I can be an affiliated to there, and I can actually get an outreach to a large number of other colleagues from different domains who have the knowledge and know-how for dealing with large number of problems, which can be used also to solve problems which are pertinent to precision health. So that's how we developed this one of the vertices over there in the 8th school. And the idea is how you can develop methods for non-invasive stratification of people for preventive health we can integrate knowledge systems from Ayurveda, yoga, different kinds of systems, because today's day a person wants optimal health, and it doesn't matter to him from where he seeks it. But it is a problem that if he keeps on seeking too many health options, maybe that is also might be counterproductive. The country is moving towards everybody eating millet, but now millet, how much to eat, who can digest what kind of millet, all those things. So there are generic recommendations which are needed for everybody, and they need to be customized or they need to be contextualized from a person's makeup, is what we are going to talk here about, right? So I this is a meaning I like doing mind mapping, and this is a kind of mind map. If we were to delve on today's workshop, first we need to ask why there's a need, and whether people are practicing, and it's been there for so many years. But why you need to have Ayurgenomics like framework in place, and can that allow us to bridge Ayurveda with modern medicine? How do you understand the concepts, and how do you integrate it with today's sciences? And then how do you evolve a framework which can translate the contexts of Ayurveda, and then how do you take it forward? So see, these are the things which I would be discussing. So the first aspect is, why is the need? If we need to have a field to be developed, we need to have stakeholders of every meaning every people who are participating. So is there something for the modern people to to kind of think about Ayurgenomics is needed, and would Ayurveda people also benefit by having such a system? So this is the first thing. So we will address this the first. Why we are worried that we need more methods. And this has come out of the WHO's global disease study, which ended in 2017. There was this document which talked about large number of disease from different countries and huge number of data sources. And what came out very significantly in all these studies is that even though we've been able to manage to live longer, if we've got longevity, but in most of the disease, the last 10 years of our lives, we are actually living with certain disabilities or not so good health. So the health longevity has reduced by 10 years, even if we are living longer. And this contributes to a large number of deaths, large number of lost life years, and majority of comes from non-communicable diseases. So the need of the hour is how you can scale developments, how you can have a personalized and preventive approach so that you are able to reduce the health burden. The other things is that there has been a lot of shift in cultural practices as we are moving. Rural places are having urbanization. Rural people are moving to transitioning to other things. Urbanites are moving to different kinds of lifestyle and eating practices, and all these are actually contributing to a large number of prevalence of many kinds of diseases. That's because sometimes we are ancestrally, our body or our genetic makeup are adapted for certain things, and if we shift or transit to a new environment, the maybe we might not have the genetic wherewithal to cope up with those altered environment. And if we know our genetic makeup and our environment in a much better manner, then maybe we would be able to calibrate, because it's not that we will stop moving. That's not the way. But how do we move? How do we transit? And how do we understand ourselves after transition is what is more important. This is a classic example of lactose intolerance. Earlier, we were hunter-gatherers, and then we moved to dairy farming. So lactose digestion was needed for a longer period of time. The populations which were ancestrally hunter-gatherers, they still have lactose intolerance, but the majority of the populations are not so lactose tolerant. So there's this book which is interestingly written by the name called Survival of the Sickest. It's a very old book, but it talks about how many of our conditions of our present diseases are because we are not in sync with the changing environment, and there are many examples there. So one of the classic example is domestication of wheat. Wheat has become polyploid, and it has led to the increase of celiac disease just because we needed to make wheat which would not shatter, and in the process we made more gluten from the same wheat. And if people have started to move into gluten this diet, then non naturally. So that's why, like arid desert region, millet was the traditional food, and people are now moving to wheat, and that is leading to celiac, that's leading to anemia. So there's a major push over there that how we can actually bring back millet into people who are normally, and millet does not also require so much of water to grow. So the whole environment and eating and our culture, how they are so intricately connected, and if we do not address them in totality, then there's a possibility that we might actually lead to a large number of mismanagement of health of ecosystem and people. The other thing is there's been a very major focus on wellness. There are wellness industries which are growing uh, 10 to 20% in a year, and a large number of people want to kind of live with good health, and focus is future is mostly, most of us now are wearing apps trying to monitor how much water we drink, how many steps we take, and this is all the we can ourselves see during our time how much we have become much more concerned or mindfully concerned for wellness. And one of the major things is that COVID has been a teacher in more ways than one. And it's been a leveler in the area of modern medicine and Ayurveda. It's been a great leveler because each of us who battled the COVID had a very different story. And each of the family member also had a different story. We realized that with the same virus, all of us were not impacted to the same way. Some needed oxygen, some had a inflammation episode or a crisis. Some were not so infected, some were repeatedly infected even if they had vaccination. And there were infodemic of alternatives like so you have this, you have that, and even the modern medicine was also kind of trying to get solutions in a very hit and trial method. So something was recommended, it was pulled out, then the next thing was recommended because it was something very new. But this time, to Ayurveda, it was business as usual. Because for Ayurveda, every day, if you can think about, is like a COVID pandemic situation. You get modern descriptions, you get modern tests, modern reports, and the Ayurveda doctor has to look at those reports, connect to their Charak and Sushruta, and evolve a medicine. So for them, every day is something like how you are battling a COVID pandemic. And actually, pandemic is described in Ayurveda. It's called Jan Padudhvans. I don't need to tell this audience, but it actually has a vivid description that even though there is dissimilarity in constitution of human beings, there are factors which are common to individual, and initiation of these factors lead to simultaneous manifestation of disease, and factor which can actually lead to destruction of countries. Factors which are common for all inhabitants are air, water, locations, and seasons. Imagine this is being told 1500 years back. So it's not that what we are seeing is disease of today. Many people have asked me, you mean you are working on Ayurveda, but there's climate change, there's a season, the geography is also changed, the diseases are new. It's not so new. We talk about genomics and we say humans moved 100,000 years from Africa. Ayurveda is only 5,000 years old. So in that context, it's not really old, and people would have been seeing patterns, and people would have been not sitting idle for so many years. It's only we think that today only we are doing all the developments, but maybe we don't know the methods what they use and how they use and what is their language. I just did a survey because I wanted to know how people have become aware after COVID, and turns out a large number of people, though still they still do not know whether Ayurveda is practiced by a practitioner. They might not have seen a registered Ayurveda practitioner. They don't know whether using herb like haldi is, I find, consuming haldi, does that become Ayurveda, or is it just you are taking a spice? That differentiation is not there in people, but nevertheless, large number of people got aware that Ayurveda is something we can also go for answers in conditions such like the COVID pandemic. Now, this is the need. There's a greater need in many contexts. Now, how do we address that need? So, some of the people have been in the modern context have also been worrying about this, because we are all anticipating, could there be better ways to tackle diseases? And that's where the genomics come. And genomics is so beautiful because this is one unifying language of chemical letters, where all life forms are in the same language, be it bacteria, be it human, be it plant, be it mouse, chimpanzee. Everybody has DNA as their language, and this is what was propounded by Watson and Crick way back. I just thought for putting this audience in a focus. All the cells have these chromosomes, and these chromosomes, if you open them up, there's this DNA, which is the book of life. Like you have letters, like you have alphabets, you have this DNA as the chemical letters. If you thread them together, just like words, then you get something called codons. You thread the codons together, you have genes, something like sentences. You thread the sentences, you get chapters, which are chromosomes, and you put all the chapters in a book. It's a genome. So we inherit one such book from each of our parents, and the science of understanding this book is what is the science of genomics. Now, this book of life is not same for all of us. Just like if you can think you got a book and you have to retype it, you will make errors, right? So the moment you make some changes in the DNA, then that DNA will change the codon. That codon will change so the DNA is not the message per se. DNA is transcribed into some message which is called as the RNA, which is a process called transcription. And after transcription, three letters, like I said, the codons are read, and you incorporate an amino acid. There are 20 amino acids, so the combinatorial of these amino acids will make a protein, and which will be dictated by what is the DNA sequence. So you have one change in the DNA, RNA will change, and that will incorporate a different amino acid, you'll get a different protein. So sometimes these changes can cause disease, like sickle cell anemia. Sometimes it can just make us different, right? So, and this is worth remembering, that we have 10 power 14 cells in our body. Each of our cells have these chromosomes, and each of these but express different parts of the chromosome. That's why we get different tissues, and this is where our individuality develops, right? So you can think about it now, and I'll come to that, is that RNA could be equated to doshas, whose preponderance is different in different tissues, and that is what gives a tissue individuality, and overall also it gives a human individuality. So you have the same dosha described for a person, same dosha described for a tissue. So your currency is dosha, genomics currency is RNA. So that's one interface where we can start to think about, could we start to compare them, what it means at the genomic level when you're talking about dosha. So way back, many years back, people thought that if we know read the entire book of life, then we will know what is the book, and then we can compare that book with all other books and try to know why this person is diseased, why this person is different, and things like that. it was easier said than done, because when we ended up sequencing the human genome, we ended up finding that not only humans are very, very different at the genetic level. Between relatives, we have 1 million differences. So you can imagine the combinatorial possibilities you have. And these also propelled different areas like the metagenomics, where we got to know that we have more number of bacteria in our body, and that also came with its own gene. So if humans give 20,000 genes, the bacteria which resides in us give 200,000 more genes. So now we become a meta-human. We are working with microbes all over us also, and these microbes are not randomly all over us in different parts of our body. You have different kinds of microbes, and different people have different proportions of those microbes, but overall in certain parts they are colonized by similar kinds of microbes, because microbes need a substrate to grow, and bodies fluids will provide different kinds of substrates, and that's why you will get different microbes. So now life has become even more complicated, because 10 years after the human genome got sequenced, people wrote essays that we still have a very, we don't understand what is individuality. We cannot tell from the genome sequence who is a normal human. And even medicine doesn't describe health in the absence of disease. So you're not diabetic, you're healthy, you're not asthmatic, you're healthy. But from the health state, how healthy you are, those baselines are really not understood, right? So at this point of time, people are still deliberating, how do you define individuality? What does a normal human look like? Then there are converging themes in diseases. For example, if anybody is cell biologist or anybody does biochemistry, you measure the same biochemical parameters in all the diseases. But one person has diabetes, the other person has cardiovascular outcome, but you are measuring wound healing, you're measuring inflammation. So why doesn't the person get all the diseases? So it's like now people know that in a disease, the outcomes are common, intermediate pathophenotype, and those pathophenotypes are signatures for how the disease is going to progress. And those are the ones where people now need to know, do different people have different levels of intermediate pathophenotype, because of which they are going to progress differently to diseases, or they would be differently healthy. And now the whole thing has come to this converging theme in diseases, where gene environment work in conjunction through these intermediate pathophenotype to give the diseases which are important for understanding management. And if you know that if people have differences in these axes, then would you be able to arrive at individualized management? So this is where we are moving towards predictive, preventive, personalized, and participatory. People call it as P4 medicine. The other thing is, now when genomics has liberated a large number of areas, so people from different areas started working on genetic basis of different diseases. Some would work on diabetes, some would work on asthma, some would work on cardiovascular disorder, and then we ended up with this kind of a mess. The same genes are involved in two diseases, and so there are diseases which are connected to the same gene. Multiple genes are connected to same diseases. So there is a whole lot of systemic kind of connections between diseases. And you have multiple sclerosis, you have rheumatoid arthritis, all of them have an autoimmune link, right? So the genes might also be shared for autoimmune diseases. But we've been in the modern medicine looking at an organ specific system, and that's why we tended to treat them in different pockets. But once the genomics came, all these seems to be connected. So now there's been a move to a systems biology perspective. The other paradigm which has got shift is also the medicines. So there is this site which has been made by doctors themselves. It's called NNT. It is actually an outcome of a lot of Cochrane trials, where it's basically information resource for doctors to understand when they prescribe a medicine how effective it is going to be, right? So this is called as NNT site, number needed to treat. And this is typically, supposing this is a drug for schizophrenia for a particular drug, or this is a drug for heartburn like omeprazole. The idea is if you are treating so many people, how many people are having an effective outcome? And that's a term which is given as NNT. So in this, 50 people are treated, only one person is effectively treated, then the NNT is 50, here NNT is 5. So the larger the value of NNT, the lesser the number of people are getting treated. And this is just because all the drugs are primarily gone through clinical trials, but based on population averages. When you deliver it to a population, there is inter-individual variability, because of which not everybody has an effective outcome. And once you know that, and this is appreciated. This is appreciated in clinical settings. Any person who has diabetes has metformin. Everybody has a different dose requirement of metformin, different time requirement of metformin, and different response to metformin. So now move has been to a one-person trial, where the idea is a one person himself or herself becomes sometimes a case and sometimes a control, an aggregate of that across different people are likely to give you much better outcome estimates. So there's a gap in knowledge. The gap is that we are mixed population, and if we are supposing have ways to segregate this population into maybe homogeneous groups, then in that case, would we be able to reduce the NNT from such a large number to a subset, so that you are able to not cause harm to a set of people and more effectively treat a set of people. So NNH is the other term, which is number needed to harm. So you these numbers are all there in the NNT side. So and the other thing is what people are now talking about is a human is not a static thing. Like coming to NCCS from Madam's house, which I took 15 minutes to 1.5 hours maybe in a particular day, and that's because everything is changing, right? So you need to have a GIS mapping, and similarly, the human exposure is changing. Yesterday I was in Ahmedabad, today I am in Pune, before that I was in Jodhpur. All the exposures is changing, food, everything is changing, right? So now what people are trying to do is, if they can capture a person's gene, the environment and the temporal variation, and do it in a big data. So you keep on capturing a person's information, and then try to see what are the kind of networks a person has, and from that network would you be able to bin them into different groups, and that way you will be able to identify who are the people who respond to a particular condition. The US is taking this route, and this is a very big data route. You take millions of people, you capture all their information, and you start to aggregate the knowledge, cluster the information, and try to see what patterns come out, right? Now, such a kind of thing is like, I tell people that it's a puzzle of Taj Mahal. You don't tell the person it's Taj Mahal and you give the puzzle to be solved. They'll take years. They will eventually be able to do that, but you don't have a scaffold of information, right? So you are not able to kind of structure the information. Now, this P4 medicine, what I talked about, is actually not new to India. Nature, actually this was way back in 2016, they came up with a special issue, and in which they really acknowledged for the first time that Ayurveda medicine was laid out in Vedas, and the treatment is tailored to a person's constitution. Maintenance of health in healthy and disorder and disease is something which is actually talked about in Charak in Sutrasthan 1500 BC. So all the things which we've been talking about, and maybe 2 decades back, we read about this, and we thought that if modern people are also moving to this area, can we actually integrate the 2 sciences? And that's where there was the genesis of this area. And we've been working with Bhavna, she's an Ayurveda doctor, for the last 2.5 decades. We developed, and there were lots of team members from different areas to develop this thing. So why is Prakriti relevant? Because P4 medicine we talked about, predictive, preventive, personalized, participatory. An Ayurveda doctor assesses a person in all the dimensions: physiological, anatomical, physical, psychological. to be able to arrive at this multi-system composite phenotype, which is called Prakriti. And it remains, the interesting thing about it is it remains invariant throughout the life of an individual. It predicts the person's likely response, likely disease trajectory, and allows them to manage the disease. So, and any population in the world can be pinned into 7 broad types. Not that everybody will be, and it's a spectrum, but at least you are able to divide the health into 7 broad axes of variation. So that's a good start, right? Now, how that is, so this is just for this audience maybe does not require, I just for completeness, I'm trying to this is the way I also understood how to go about starting Prakriti. So just like RNA, Tridosha is a common organizing principle, and everybody will have these 3 physiological entities in them, which and Ayurveda is called as Vata, Pitta, and Kapha. For our language perspective, it would be the kinetic and the metabolic and the structural component. And an individual will be in any of these vertices. So if it is more towards this, they have more Vata in them, more Pitta in them, more Kapha in them. So individuals who are of, let us say, Pitta type, any movement to this direction will be a disease for this, but is a health state for this. So the way you will describe the disease state will be actually perturbation from the person's homeostatic state, which would be different for different Prakriti people. And getting them back to homeostasis will also be different, because it will be to bring back the person to that person's homeostatic state and not everybody to the same homeostatic state. And this understanding of this dosha is what leads to this concept of something called Trisutra, where you can ascribe the causes of through Vata, Pitta, and Kapha, the phenotype also through Vata, Pitta, and Kapha, and you also design the therapy based on which has got elevated, and as a result of which the drugs are also defined on the basis of the dosha presence. So this is the unifying organizing principle which links the two. And also it was very interesting to me, and this was the place where was my aha moment, because in Ayurveda there are shlokas which they get translated to Vata, Pitta, and Kapha, you actually start to see terminologies which you see in the biochemistry textbooks. For example, Vata governing cell division, proliferation, signaling, movement, transport, cognition, excretion, initiation are all governed by functions of Vata. Pitta governs these functions related to metabolism, host surveillance, and Kapha governs the functions related to shape, immunity, strength, storage, etc. And the most interesting part was that it also described that an individuality can just not be described by a parent's genetic component, which they call as Shukrashon in Sanyoge, but also the mother's prenatal environment, which is called Garbhashay, Mathur, Ahar, Vihar, Prakriti, and then is impacted by all these things which are in modern our sciences. Each of them are different subjects. So it is governed by ethnicity, Jati Prasatta, what runs in the family, Kul Prasatta, what geography you are coming from, Deshanupatini, what time you are observing, Kalanupatini, what age you are looking at the person, Vayanupatini, and then Pratatmanyata. So an individual is not a consequence of that one person at one moment, but it's contextualized from all these perspectives. And that is why it's like a much more deeper understanding of individuality than we understand. But then that sounded very abstract. If you have such kind of individuality, how does that confer or convert to a person's Prakriti? So in Ayurveda also, just like we have for RNA, there are seeds of where Vata is more, where Pitta is more, and where Kapha is more. So whichever tissue they are impacting, that tissue individuality will be governed by those doshas much more. So if an individual is of Vata Prakriti, then their Vata tissue might have much more differentiating feature, because they would have inherently elevated levels of Vata or elevated levels of Pitta. And that's how this then becomes a window to giving an individuality to a person, whether the person is Vata, Pitta or Kapha. And that's how you get to the multi-system classification, that whether they move fast, talk fast, walk fast, digest better, those are all governed by those enrichment of those kind of doshas in particular tissues, which we give those phenotypes. And also many diseases are described for people who have elevated Vata, elevated Pitta, elevated Kapha. Some of them can also be described by today's language. The most interesting thing is that how they arrive in today's time, having all these descriptions, how they arrive at how to design a therapy. And that's also described by a term called yukti. There's more to it. I'm not an Ayurveda person, but this is something where it actually tells you that how you can assign a person, meaning you get COVID, how you evolve a therapy for COVID. You have to have a reasoning to be able to arrive at that. So I see Ayurveda and modern medicine, there are differences. Reactive, proactive, but treat molecular imbalances, so you know which molecule is being treated. Here, it aims at systems perspective, more of a reversal. There are specific molecules, there is polypharmacology here, there is holistic and individualized component. Modern medicine is reaching towards that. So how do we bridge this? If we can actually build a bridge between both of them, then we can get an objectivity and today's terminology, contemporariness, scaling, and also globalization, if we are able to achieve to link this. So that's where, how do you bridge the gap? How do you understand the concepts? And that's where we started in way back. We are nowhere near, but I'll give you a quiz just to break down my monotonous dialogue. If I give you these terminologies, what does it strike to you? Can anybody tell? The students, can you tell? Is there a common thread to all these terms? A, all of them start with A. Any vote taker? Nobody? Please. So I've put you to sleep, is it? Can you just tell? Chemistry. Okay. Some other thing? Color. Okay. All of these are colors. These are these colors. But if I give you this number, will you be able to make this color? I will not be able to make this color. What would you be needing to make this color? But I could make it, and I could make a different shape also, right? So what did I have with me to be able to do this? It's simple. I had the RGB codes, and if I have an RGB codes, then irrespective of what digital display I have, I'll be able to arrive at that same color, and I'll be able to make a different shape. So now Ayurgenomics is this, that something is written in a language which we do not understand, and we want to achieve some place. So we will have to get it to a RGB language. And the language which I thought we could integrate is the language of biology, or the language of biochemistry or molecular biology. So that's where we developed a working hypothesis that if people are of Vata, Pitta, and Kapha, if we take individuals who are at the extreme, and Ayurveda describes them with different susceptibility, different biological readouts, then will I actually see differences in them? So that's how we developed this working hypothesis. How much time do I have? Because I have a second. 8 minutes, okay. So that's how we developed our first working hypothesis. We decided that we will take people of a particular endogamous population or genetically homogeneous population, classify them on the basis of Prakriti, and ask a question: what do I see? Do I see a difference? Okay. And this is what we did. We were funded in an Indo-Russian. I will not go into the history of why Indo-Russian, but nevertheless, we were funded by DST and we started this exercise. So during that time, I was part of an Indian Genome Variation Consortium project. So we know what is the genetic map of India. So we assured that we take one particular population, because you realize this was a time when everybody had a very, very negative opinion about getting into this research. So if I say there's a difference, they will say you've taken people from different parts of the country and saying they are different. So we needed to be much, much more kind of rigorous in whatever we are doing. So we actually took populations who are more related to each other at the genetic level, and then went around screening them by a questionnaire which Bhavna had developed, and identified people who are at the end of the spectra, and asked a question by routine biochemistry, by genome-wide expression study, do they differ? And first thing we found was, even the routine biochemistry where there's a range, even within that range, Vata, Pitta, Kapha had a different range. So that means you wait for, so if supposing this is a parameter for, let us say, a lipid profile or a hematocrit, then these groups will actually be within a range. So now you can realize that movement of this to this will be a disease range, but we wait for this to move till this, till we tell that that person is diseased, right? So like that, there were different parameters in which they differed, and these were broadly, for example, fibrinolysis, prolactin, they were more elevated in Vata, hematocrit were more elevated in Pitta, lipid profiles were more elevated in Kapha. And then there were processes with gene by expression studies. We found they were different in different kinds of people. Now this was very interesting. Cell cycle came up in Vata, and there's a shloka in Ayurveda, which is Parmanu Bhed Na Pari Sankhya, work of Vayu, which is also there in our supplementary. But then we asked, this was peripheral blood. So this was the first time we could actually tell that there are ways to query doshas through genomics, and there are molecular correlates, right? So 10 years later, we thought that do they have a cellular phenotype? And can we actually recapitulate Vata, Pitta, Kapha, Prakriti within the cells of the people? So we took Vata, Pitta, and Kapha, Prakriti individuals, same age group, same gender, same ethnicity, same genetic background, made their cell lines, basically to see whether there is a cellular phenotype. I'll cut a whole story short, but one of the things which we found was that the rates of cell proliferation were very fast in Vata compared to Kapha. This was done by all the state-of-the-art cell cycle assays one can do. So then we asked a question, if cell cycle is fast in them, what happens if you give, supposing, a damaging agent like UV? Now, when we give UV, these Vata cells, and these are like multiple biological replicates and multiple technical replicates, we found these observations. So when we do this, we give UV, then we find that there are more dead cells in Vata compared to Kapha. And so the numbers dwindle quite a lot in Vata. But if we wait for them to recover, after a point, all of them come to the same number. And that's because the surviving cells in Vata have a growth advantage, which actually allows them to come back to the same number. This was something which we published in a journal called Cell Cycle. But then we wanted to ask, is this relevant? Is this, why is this so? So we found that in ENHANCE, people have been working with bipolar disorder, where there's a lithium response. 45% of the people do not respond to lithium chloride. They were also trying to do a similar kind of an experiment where they had lymphoblastoid cell lines, and they were wanting to look at differences with lithium responsiveness. And they ended up finding that the ones which were lithium responsive actually had a cellular phenotype which was similar to Vata, and the other one was more Kapha. And it was very interesting because NIMHANS has also now put up an integrative medicine place where they are trying to merge the sciences together, and they are also looking at these ways. Now, why this becomes important? This is like a cell proliferation. We did not know how Ayurveda doctors looked at cell proliferation, but nevertheless, we now have a modern cell proliferation phenotype, which is linked to certain aspects of disease response, and even Alzheimer's, as I understand from them, has cell lines are much, much higher proliferating compared to the normal cell lines. So they can actually find which cell lines of theirs are contaminating their cell lines. So that's what Dr. Biju from NIMHANS told me. Now I give you a quiz to all the Ayurveda doctors. You see, there's this patient A. Normally, they will approach an Ayurveda doctor and tell that they have a disease called Fanconi anemia. And you look at this person and you see all these kinds of phenotypes. Okay, what will you say this person is? What kind of Vikriti? Can you tell fast? I don't have. Hello? Huh? Vat Prakriti, right? He has answered on behalf of all of you, and
The Vice Chancellor.
absolutely right. It is Vat Prakriti, right? And so you will have a therapy for them for treating the Vat, right? Now, if I say that there is a gene associated with this, which is called FANK-A, and the gene's function is for cytogenetic instability, hypersensitive to DNA, increased chromosomal breakage, DNA repair, it actually goes on to intersect with similar kind of things. And what I did was I did a query in human phenotype ontology. I said, give me the disease which has abnormality of cell cycle. It gives me Fanconi anemia. I show the feature of Fanconi anemia to an Ayurveda doctor. They say Vat, and we also see cell cycle proliferation is a readout. So now this is the thing which I am trying to emphasize, that we need an interoperable platform where I post something to an Ayurveda doctor. He comes out with a terminology which also makes sense to a modern person. So why can't people sit together and work with a common language to be able to integrate these sciences, right? So this is where I will stop here, because I think I'm short of time, and I'll talk about the more applications in the next part of my talk after Supriya gives the talk. So I will deal with this in the next one. But this is where I think Ayurveda has a lot to offer in terms of making a language interoperable, and in terms of if we just need to keep our mind open and we need to acknowledge the merits and demerits of each of the system, get them across on a table and not become defensive from both the sides. So I'm trying my best to develop that kind of a platform where all of us are comfortable, and even we benefit as molecular biologists, large number of insights we can get. So I will stop for now. The next part I will deal in the next half of the lecture after Dr. Supriya gives her talk. This is basically my mission was achieved when he says that it is Vat and it's a cell proliferation, right? So thank you all for now. Anybody has any questions? Yeah.
Work is being done on DNA methylation, which is related to the chromatin regulation. How do you look upon at the correlating with the Prakriti?
So yeah, so I think DNA methylation, microbiome, these are more important aspects which will be very pertinent when we start to have querying Ayurveda in integrative medicine setting, because people are now finding that a lot of lifestyle transitions are also related to epigenetic changes in them, right? And Ayurveda, lots of therapeutic procedures are also to do with reversal of the disease. So one way you can do the reversal, you cannot change the genes of a person, but you can modify or modulate the environment. And one of the ways by which, after modulating the environment, the expression changes is through epigenetics. So the signatures of modulation would be apparent in epigenetic changes, which could also be visible in expression. So you can use the expression as a handle and epigenetic as a mechanism for the changes.
And a lot of work can also be done with the HLA typing, which is again correlated with the market.
Yes, Dr. Bhushan Patwardhan had initially done a work on HLA, but HLA field is it has so many variability that one needs to first understand the HLA classification itself then correlate it. But actually would be very important because we found that the axis of immune response is very, very important in describing differences between Prakriti type. With Prakriti have much more higher inflammation linked immune component in them. Vata have lower immune response. So these are all really, really pertinent. Yes.
Very nice talk, Mitali.
Thank you.
And you have done very good studies on the cell proliferation with all the Vata Pitta curve. So you saw that in a curve of Prakriti, the cell proliferation is
Slow.
slow.
So
So is it related? Suppose you are treating all the three different types of patient with some Ayurvedic drug. Do you see there is a slow reversal or
so
no
I
reversal in
have
cough?
I have not gone that far. This was a
Okay.
very preliminary study, but this opens up these questions, right? See, normally what happens is I've been telling this to my colleagues that they take cell peripheral blood to prepare monocytes and all culture for doing some challenge assays, right? They will have a habit of going to one or two persons to keep on taking their blood, and some they will say, this person's blood, my experiment never works. Right. So because many times you really don't make those observations of inherent differences. Once you start making those observations, even any cellular assay, you will start to see that there are actually differences if you do not work on pure cell lines or the immortalized cell lines, you work on people's cell lines, you might start to see these differences. And convincing cell biologists, believe me, Vivek Natarajan was there who did help with the experiment. It's a very, very long order for them to take Ayurveda and do a cell biology experiment, meaning it takes time for people to get convinced. This kind of paper, if we do not publish, so the thing is that now it opens up a whole lot of things, that if you have Vata cell line, Pitta cell line, Kapha cell line, you make a CRISPR mutation, same mutation in all these cell lines, and see how they progress. Or you give a different challenge to these cell lines, or you give the different drugs which are for Vata, Pitta and Kapha, and see how they. So there could be so many more outcomes out of this. My hands are tied. I'm not a cell biologist, but I wish people take it up, and these would be really wonderful.
Thank you.
I think in the interest of time, maybe we can do that later. Yeah. Thank you so much.
Thank you for that wonderful, insightful session. All of you must have read Da Vinci Code, the novel. I could remember Professor Langdon while she was talking. She was decoding so many facts. Ayurveda for us, amhalas navane kaala. So thank you, madam, for this wonderful, insightful talk. And now for the next speaker, I would request the Professor and Head of Department of Ayush, MUHS, Dr. Carly, madam, to introduce our next speaker, Dr. Supriya Bhanirama.
Good morning, all. This task of introducing our guest speaker is a great privilege for me. Dr. Supriya Bhanirama. She has completed her MD, PhD in Ayurveda, MA in Sanskrit, and two diplomas, two PG diplomas, one in research and second in bioethics. Since 2013, means since last 10 years, she is working as a scientist and head of Metabolic Research Lab, Interactive Research School of Health Affairs, Bharati Vidyapeeth. She has completed more than 25 funded projects, including government funding. She is a member of Scientific Advisory Committee for ICMR. She is a member of Expert Committee for Product Approval of Food Safety and Standard Authority of India under the Government of Health and Family Welfare. She is a member of Data Safety Monitoring Board for many Ayush interventions related with COVID-19. She has delivered more than 350 lectures based on research, and she has published more than 50 articles in national and international journals. Now, ladies and gentlemen, please help me to welcoming Dr. Supriya Bhalera.
Okay, good morning, everyone. And before the slides start, yeah. Am I audible now? Hello, am I audible now? Yeah. Okay, so good morning, everyone. And first of all, I would like to thank VC Madam for giving me this opportunity. And I always feel proud to be a first PhD holder of NUHS. And in that capacity, I am always happy to be on the NUHS platform. So with this, I would start my presentation. And the topic which was given to me was understanding Ayurveda in the light of genomics, applications in integrative health and personalized medicine. Slides are not moving. Here it is done. Okay, so by the time slides come, whenever we talk about Ayurveda, it is not only about formulations or therapies, but as Dr. Mitali has nicely said, it is more about the concepts of Ayurveda. And when we talk about concepts in Ayurveda, Prakriti is the foremost. What I am going to highlight in my today's presentation is 3 aspects of Ayurveda. One from concept, that is Prakriti, then Triphala as a formulation, and Basti as a procedure. These 3 things I am going to highlight in the presentation today. So as Dr. Mitali has already explained, and we all know that Prakriti explains intra-individual variability in the health states of individuals. And this individuality comes from the relative proportion of doshas. Prakriti is nothing but the measurable and observable phenotype of measuring these proportions or measuring this relative quantity of dosha in the body. And as Dr. Mitali has also pointed out, Prakriti is responsible for response or risk of an individual for a particular disease, health and disease trajectory, and then management of food, medicine, lifestyle in that aspect. So basically, what is dosha? Because we said that Prakriti is relative proportion of dosha. So what is dosha? They are the common organizing principles as per Ayurvedic philosophy, and Vata is kinetic energy, Pitta, kinetic entity, Pitta is metabolic, and Kapha is responsible for structure. These doshas govern all physiological functions in all tissues of human body across all levels of organizations, and they work in conjunction. They govern shape, functions, response, perturbations, everything. So this is how the doshas are, and they define Prakriti. the main functions of doshas. Dr. Mitali has already shown this slide, and also the clinical features of Prakriti as per the predominance of each of the doshas, Vata, Pitta, and Kapha Prakriti. So these descriptions also are very interestingly described in Ayurvedic textbooks. So what I'm going to share with you is my experience in Prakriti research, and to tell you that though I'm an Ayurvedic graduate and postgraduate, I was little anxious, or I was not very sure that what I will do, what I will get after doing this research. So I started my research on Prakriti in 1999, and we did a lot of Prakriti related epidemiological studies, and then we further went on to do some Prakriti and cellular markers, and then from 2010 to 2013 we did some Prakriti genomic studies. Now you will be confused like what after 2013, that answer I will give you later. But we started with some epidemiological studies, and we tried to find out, like, as it is described that Prakriti is responsible for disease risk. Actually, do we get that kind of information? Actually, is it a sensitive tool to identify the differences? And when we did the Prakriti analysis of hypertensive patients, we found that Pitta Prakriti individuals were more prone to, or were more in this cohort. But when we did the similar study in epileptic patients, the proportion was completely different. All three Prakritis were seen to be equally affected by epilepsy. So this was like, okay, at least we could get the differences. There was a difference in Prakriti types in hypertensive patient and difference in Prakriti types in epileptic patients. So this was good thing, though there was an distribution of all three Prakritis in epilepsy. We got a hint that yes, there can be differences, and this is like how Prakriti work as a non-modifiable disease, non-modifiable risk factor. We further analyzed the etiological factors in these epileptic patients. And if you could see here, in Vata Prakriti individuals, the yellow kind of proportion is seen more. That represents atrophy as a physiology, and that was seen in more. That was seen more in Vata Prakriti patients suffering from epilepsy. Whereas in Pitta Prakriti individuals, the second upper, and low, from lower side, the second bar or second proportion is higher, that represents hemorrhage. So Pitta Prakriti individuals, the etiological factor was found to be hemorrhage, and in Kapha Prakriti individuals, the lowest bar, which represents tumor, was seen as a more pathological factor for epilepsy. So this also goes, my Ayurvedic colleagues know that this goes in the philosophy, or what are the basic concepts which are given in Ayurveda, that in Kapha, the tumor is seen as a pathological factor, in Pitta hemorrhage, and in Vata atrophy was seen as an epilepsy factor. More interestingly, when we studied the response to epileptic drugs in different Prakriti individuals, and these are modern anti-epileptic drugs, these are not Ayurveda anti-epileptic drugs. I got privileged to work in modern hospitals like KEM Hospital in Mumbai, and then Nair Hospital in Mumbai. So there we did this study, and almost in 565 epileptic individuals, we observed that response to anti-epileptic drug was more. The responders were more in Kapha Prakriti, where non-responders were more in Vata Prakriti. That gives us a guideline that if a Vata Prakriti epileptic patient comes to you, then you have to arrange frequent monitoring. The therapeutic drug monitoring has to be done very carefully in Vata Prakriti individuals, because they respond poorly, they require dosage change, they require drug change more frequently as compared to other two Prakriti individuals. Going further, we also studied Prakriti-related biochemical markers in a same disease, in a same disease entity. Whether Prakriti-wise clinical manifestations differ, the biochemical markers differ. And we did this study in obese individuals, and we could see that in Kapha Prakriti obese individuals, serum cholesterol, LDL cholesterol, carbohydrate, they were on higher side. Whereas in Pitta Prakriti obese individuals, triglyceride levels, LDH, cortisol, these levels were on higher side. This gave us again a link or hint that Kapha Prakriti individuals may be more prone, Kapha Prakriti obese individuals may be more prone to diabetes kind of diseases, whereas Pitta Prakriti individuals may be more prone to cardiovascular diseases. So this was also study done in almost 60 individuals, each of Kapha Prakriti and Pitta Prakriti obese. We further went on and we did cellular level differences. We studied 3 cellular level differences in Prakriti: platelet aggregation, neutrophil phagocytosis, and monocytes proliferation. This work was published as a and submitted as an MSc thesis to Mumbai University, but we could publish only platelet aggregation related work of this. And I know that cell biologists are sitting in front of me, so I will not go into much details of this. But we studied the ADP-induced platelet aggregations in response to aspirin in case of platelet aggregation, percent phagocytosis and phagocytic index, response to forgolumeric acetate in case of neutrophil phagocytosis, and in case of monocytes proliferation, this percent viable cells and response to PHA. So these were our parameters, and I will first explain the results related to neutrophil phagocytosis and monocytes. That in case of phagocytosis, Vata Prakriti showed lowest phagocytosis and phagocytic index both, and both these parameters were maximum in Pitta Prakriti individuals. Similarly, response to PMA, though the phagocytic index was lowest in Vata Prakriti, response to PMA was maximum in these Prakriti people, and it was lowest in Pitta Prakriti. In case of monocytes proliferation, we saw that response to PHA was highest in Vata Prakriti individuals at higher concentrations. So these were some preliminary studies we did on the cellular responses in Prakriti. We could not publish this paper because of some limitations, but I am happy that after 10 years almost, Dr. Mitali and her group presented like how cellular differences can be reflected in Prakriti. I mean, Prakriti can be reflected even in cellular differences, cellular level differences. But these were our initial attempts, and this was quite interesting that how the baseline things differ and how the response to different agents also differ in different Prakriti types. Coming to platelet aggregation, the paper which we have published, we have seen that maximum platelet aggregation was seen in Vata Kapha Prakriti individuals. Actually, when we started this work, I thought that Kapha Prakriti is responsible for atherosclerosis like events. So maybe the aggregation, and it is responsible for Kapha dosha, is responsible for structure, storage, stability. So I was hoping, or I was expecting, not hoping, that Kapha Prakriti individuals will show maximum aggregation. But we found that wherever there is a bar, the maximum platelet aggregation was seen. And interestingly, the other thing was response to aspirin was poor when there is a Pitta component. Now, clinically, this becomes a very difficult picture because aspirin, Pitta Prakriti individuals are not responding to aspirin. That means they will require higher dose of aspirin. Aspirin causes bleeding. Pitta Prakriti tend to have bleeding disorders. So this combination becomes very detrimental, and this gives us another guideline clinically applicable, that when there is some platelet aggregation related disease or condition in Pitta Prakriti individuals, we have to be very, very careful while managing these patients. So this was our another study. And then the logical thing was, because we could find that Prakriti and platelet aggregation are platelet aggregation differ in different Prakritis. So the logical next thing was to relate it with some genetic polymorphism, and then we chose P2I12 as a gene, because P2I12 is a gene which mediates the effect of ADP. And then we studied that whether the different Prakritis have different polymorphism frequency in this P2I12 gene. We then plotted that platelet aggregation and just overall, without classifying as per Prakriti, we could find that there is a binomial distribution of platelet aggregation. Means there were two different kind of some people were having maximum aggregation around 31 to 40, and some people maximum other group was having around 71 to 80% platelet aggregation. So we could find that there is a binomial distribution. There are two modes of this bimodal classification. There were two different clusters in this cohort which we were studying. We again saw the platelet aggregation. Again, the Vata Prakriti individuals where there is a Vata component, there was a higher level of higher aggregation seen in these individuals. This P2 gene is having three haplotypes H1, 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 H2, and H2, H2. In our population, we didn't find anybody of H2, H2 haplotype, but we could see that there were differences in the platelet aggregation as per the two available, whatever two haplotypes we could see in our population, H1, H1, and H1, H2. So H1, H1 were having lower kind of aggregation, which is the left part, and H1, H2 haplotype people were having higher platelet aggregation, which is on the right side. In Vata Pitta Prakriti, we could see that there were three individuals having H1, H2 haplotype. But you can see from these overall results that the numbers which we had were very few, and that's why we couldn't reach to any conclusion. But this study gave us some idea that whenever we are planning such kind of studies, and this was a lesson, not idea, but this was a lesson that when we are planning this kind of study, it is essentially important that to first have the idea about the frequency of polymorphism in our population, and that is how then we can plan sample size if we want to come to some meaningful conclusion. But these are our some experiences while working with Prakriti concept. We started with simple observations in OPDs, and that could also give us some ideas about how Prakriti can be a useful tool. And this whole work was done in modern medicine hospitals, modern medicine colleges. When I talk about anti-epileptic drugs, they are modern anti-epileptic drugs, and it was truly an integrative approach. And we could see that in a modern healthcare hospital also how Prakriti concept can be implemented, can prove useful for planning the management of patients. And ultimately, this is what Dr. Mitali also said, that rather than giving standardized treatments and getting effectiveness rates in different individuals in a different proportion, it is always advisable that if Pitta Prakriti persons are not responding to aspirin, then maybe we have to have some other kind of management for them, and to get 100% effectiveness at the end. So this is what is expected when we talk about integrative approach in Prakriti research and how we can go ahead. And I have said that after 2013, what we have done, so we have started incorporating Prakriti in all our research components. I mean, whatever research we do, we add Prakriti as component. And you might be knowing that now Prakriti is being researched upon by various people, the gastroenterologists, psychiatrists. Nimans is trying to get Prakriti analysis from the genetic data which they have. So a lot of studies are being done and a lot of things are happening in the field of Prakriti. And recently we have studied that Indian diabetes risk score, which is a very well-known diabetes risk score. When we added Prakriti to it, the sensitivity and specificity of IDRS is also going up. And that paper we are going to publish very soon. So this is also data from 500 individuals. And I think this is how we should integrate. And this is all we are talking about integrative approach. And when we are talking about IDRS, then it becomes a direct translation at public health level also. So this is all about Prakriti concept and how we can understand Prakriti in the language of modern science or genomics. Going further, as I have said that Ayurveda is not only about concepts, Ayurveda is also about formulations, Ayurveda also talks about procedures. So a very small thing which I would like to share, but before that, though the topic involves only genomics, now with the emergence of many omics, now we have to think about multi-omics level, and we have to do our research, or we have to scale up our research in Ayurveda on Ayurveda concepts and procedures also on these different levels of omics. And what I am going to share with you is an example of Triphala, and here I am going to talk about multi-omics. And that is a molecular mechanism of therapeutic intervention in Ayurveda. Triphala is a very popular Ayurveda drug, and everybody knows about Triphala. We have done a community survey of more than 700 individuals from different parts of country, and we found out that everybody knows about Triphala, its ingredients, it uses everything. Triphala is known to be used for in Ayurveda for obesity, and even people know that it has to be used for obesity because YouTube also features video about Triphala in obesity. But there are no mechanistic studies done on Triphala, like how it works and what exactly can be the mechanism of Triphala. This was very surprising. And another surprising thing which I would like to tell, not part of this actually talk, but when I started working on Triphala and anti-obesity, there was only one paper published on Triphala clinical study on obesity, and that too was from Iran. So we have to take some lesson from this, and we have to really gear up and publish and do some good studies on our own formulations. So going back, this work we did in 3T3L1 cell line, and that is a fibroblast cell line. And when I talk to my Ayurveda colleagues, I always say like it is fibroblast cell line. I mean, we converted fibroblast into adipocyte. It is exactly the translation of Ayurveda principle that mouse dhatu is getting converted into meda dhatu. And then how Triphala works in this mouse dhatu to meda dhatu formation. So it is not only application of Triphala, but it is some Ayurveda concept also which we are working on. So you can see that the second box above line, that is where the fibroblasts are completely converted into adipocytes in presence of insulin. You can see many red drops there. They are fat droplets which are stained by oilo-red staining. And when Triphala is treated with different concentrations, the red droplets, you can see that they are going down. So the lipid formation has gone down, indicating that adipocyte formation was inhibited by Triphala. And the same thing, the lipid content we also measured using the quantitative methods, and we could see that Triphala in increasing concentrations decrease the lipid accumulation in adipocytes. But this was not sufficient, so we also worked on the gene expressions which are involved in adipogenesis, like PPR, gamma, GLIT4, FAST, CVP alpha. Two of them are early adipogenic genes, and two of them are late adipogenic genes. And we could show that Triphala downregulates the expression of adipogenic genes in presence of insulin. So this is how we could, decode or decipher the mechanism, elucidate the mechanism of Triphala. And this is another experiment where we worked on SHSY5Y cells, which are neuronal cells. And the idea was that when obesity associated cognition is a very upcoming area. So obesity related lipotoxicity impairs the brain functioning. And there are upcoming reports in this area. So we did this in vitro model where we treated the cells with palmitic acid and caused the neuronal lipotoxicity, and then treated the cells with Triphala. And you can see that after treatment of palmitic acid, there was clump formation in the cells, whereas the Triphala treatment restored that cell morphology. And again, we did different gene expressions like FAST, that is apoptotic genes, which was increased after palmitic acid induction, and how Triphala downregulated that. The NFE2L2 is an oxidative stress related gene, and how that expression has also gone down, and TNF alpha. And the same feature we could see even in Triphala on gene expression in brains of HAFD induced obese rats. So, by both the ways, we confirm this thing like how Triphala works on the gene expression levels in the lipotoxicity associated with obesity. And lastly, I'm going to talk about the procedure, that is Basti. And here again, I'm going to show you that how Ayurveda things can be understood in the light of multi-omics. And this is probably the first study on Ayurvedic intervention where we could show that Takra Basti, that is buttermilk enema, could change the gut microbial diversity in obese individuals. And this is a procedure as per Ayurveda. The Takra Basti was given. Takra, as many of you know, that Takra is used as a anupan or concomitant medicine in with anti-obesity drugs in Ayurveda. But we directly gave the enemas of that because we thought that it is the nearest route to reach to gut microbiome, gut microbes. And that is how we gave the Takra Basti, and the whole procedure we used as per Ayurveda. The way the Takra Basti has to be given, the procedure like total 15 Basti were given. Of them, three basti were of til tail because sesame oil, because takra is ruksh or dry in nature as per Ayurveda, and there should not be much dryness. So 3 oil enema were also given. So rest of the protocol was as per Ayurveda. Only thing is we studied the gut microbial diversity, and of course this study was done with none other than Shauche sir, and we could show that after the treatment, the first grade, these are the 3 different indices which we use, and you can see in each of the case, the first bar or the red bar is of pre-treatment. The green bar represents the post-treatment microbial diversity, and the blue bar or the blue figure, the box is the plot is after 30 days after the basti treatment is over. There was no treatment between this period. So what we could see that immediately after the treatment, the microbial diversity changed due to takra basti, and 1 month after that, when we didn't give anything, the microbial diversity came to its original state. So it again gave us a guideline that if we want to have a sustainable result of takra basti, then maybe we have to increase the duration of basti, or we have to increase the duration of basti, or we have to give repeated course of basti. So this is what the implication of this study, and as I said, this is the first study on Ayurveda intervention on gut microbial diversity. So again, we showed the same thing on phyla level, genus level, and with everything we have seen the same feature, like the baseline level was exactly similar to the 30 days follow, but in between, when we studied the microbial diversity immediately after basti, it was found change. So again, the same thing emerged up. So in summary, what I would like to say that the face of medicine is changing. It is now becoming as information-driven science. Lot of information is coming up, and it is like now we have to be in pace with this information. There are systems approaches, they are talking about holistic than reductionist nature. And Ayurveda has privilege, Ayurveda has plus point that Ayurveda is always holistic in nature. And Ayurveda talks about multiple data points, which we have never analyzed also. We gather a lot of information, but actually we do not make use of that information. As I said in the beginning of my speech, that when I started working on Prakriti concept, it was my mentor, Dr. Urmila Thatte, who is a modern pharmacologist. She was she was like, no, this is something where we should work on. And I, being an Ayurveda postgraduate, I used to think like, okay, I have done my post-graduation, why I have to fill those Prakriti forms, what it is going to give me. But remember that these are the multiple data points which we collect, and they are important in some or other way. And 20 years later, when I was preparing PPT, I was thinking like how visionary she was, that she could think so many years ahead, like how this is going to be important. So this is like where we have to wake up and do something. And as Dr. Mitali said, that the science is leading, the medicine is leading to 4P medicine, where there is a prediction, prevention, personalized, and everywhere the Ayurveda role is going to be very important and participatory, where patients are also going to make choice and they are also going to get involved in decision-making process. So I already said that how Triphala story, that Triphala is very well known. We have even asked the individuals in Assam and that kind of northeast region of country, and people were aware of Triphala. So that is something very interesting, and we should make use of all these things, and we should incorporate these things in Ayurveda properly. So thank you very much.
Thank you, ma'am, for that wonderful Ha Surya, Ha Jayadrat. This is the research, this is the interpretation. So wonderful. I think both our speakers, if you go by their phenotype, as they say, they're very mild and soft spoken. The moment they take the stage and prove their work, the strength is proven. So kudos to both the speakers. I would now request Honorable Pro Vice Chancellor and you just to kindly felicitate Dr. Supriya Bhalerao, Madam. Now we have a quick session by Dr. Girdhari Lal sir, who is an immunologist working at the LCCS. I request sir to take on.
Thank you very much for putting me here, and I'm very happy to see that what's progress in the Ayurveda and the modern medicine, especially looking at the integration. Whenever we talk about integration, we have to think that integration of different branches in the same discipline and integration of different discipline in one place to have the good health. Now, what I'm going to talk about the integration of regulatory network in our body and how they actually communicate each other, and we understand that how they work and how we can interfere to have the good health. Now, what is called as Sarve Bhavanti Sukhna, Sarve Santu Niramaya. That means you be happy, have good health, and see always good. Now, if you look at in the perspective of in our country, if you want to be happy, you must be stress-free. But if you look in the context of the stress-free, the scenario goes like something totally different. So you have a lot of mental disorder, stress, and you see that the red point, which is on the bottom side, you will find that the more the financially enriched states, you will have more mental disorder and then and the stress factor. So now, if you have all these factors together and you want to be happy and healthy, I think we need to think how these things are interrelated. Now, I will put a context here. If you are chronically stressed for a long time, you will see that lot of diseases actually end up with at the end of the day. So neurological disorders are lot of stress lead to several of these autoimmune diseases. Here just some of them, including the diabetes or psoriasis, IBDs. Now question is, all of these diseases which I just mentioned here are immunologically important. Now we're talking about stress, which is neurological response, and immunological phenotype or disease, what you see, how they talk to each other. That's what we're trying to understand, how these stress factor or neurological
Yeah, that's right. Okay. So, so I told you about the conceptual concordance, but having conceptual concordance, where do you move from? How do you use this for application? So I'm going to give you some examples as to where it would be relevant in integrative medicine setting. And the first one is an example which was very old. The time when I had my first discussion with Madhuri ma'am. So this was, we had a difference at expression level by classifying people by Prakriti. So people would ask, so what? You take any set of people, you classify, you do expression, and you see differences. But we were talking about Ayurgenomics differences, and one of the things was that Ayurveda talks about if dosha differ, then those would also be kind of informative for differences in susceptibility or differences in risk. So we started to look at the genes which were different at the expression level. Were they also different at the genetic level? That is there a genetic difference which is contributing to the expression difference? So we had a whole lot of genes and we did all the statistical testing, because you need to be you can end up getting false discoveries. So based on all those corrections, you'll have to take my word for it. We landed up in one of the genes out of a set of genes, which was called as the EGLN1. Now, this gene is very interesting. This is an oxygen sensing gene. So it senses a low oxygen condition so that it will ask the body to calibrate how to cope up with low oxygen. So supposing you are in a low oxygen condition, for example, you go to high altitude, or if the cell is facing low oxygen condition, then there would be death, right? But to be able to cope up with that, you need to recalibrate your body to be able to cope up with that low oxygen condition. It turned out that this expression of this oxygen sensor differed between pith and kapha prakriti, such that the state which was there in the pith prakriti sort of predicted that it would be more responsive in low oxygen condition, and the one which was associated with kapha was expected to not to do so well in low oxygen condition. Now, when we found that kapha had an expression of EGLN1 high, which sort of predicted that it will cope up with low oxygen, and pith had the other state, we went out to look at whether this has any relevance to their adaptability to low oxygen condition. What we found that pith related genetic makeup was more present in natives who reside at very high altitude, like the pointer stopped working, I think. Leh Ladakh. So our anticipation was, if that is the case, then would it be that individuals who are of kapha genotype have more problems if they go to high altitude? And there were sojourners who went to high altitude and developed this condition called high altitude pulmonary edema. When we studied them, we found that they were more preponderance of kapha related genotype. So this, you remember, these Vatpith and kapha were done on sea level people, but we found a variation which could predict their responsiveness to high altitude condition. And then we went to descriptions of high altitude. Like I said, it has to make sense from Ayurveda perspective. So what does Ayurveda talk about high altitude? It seems there are shlokas in Ayurveda which tells that high altitude are places which are more cough path predominant. So it would be anticipated that if there is an enrichment of cough dosha in people, that they might be much more much less habituated to those conditions if they are to be adapted, meaning if they have to go immediately over there, and Pitta Prakriti individuals might be much better adapted. Now, this was something which was taken up very well by community, and because this was the first time we showed that Ayurveda methods can actually enable identification of markers which would be relevant to high altitude adaptation. Now, it's very heartening that this variation has been actually validated in multiple altitudes of the world, and after 10 years, somebody has also linked this variation to aerobic capacity. So that clearly tells that hypoxia is a very important axis of adaptability, and if you have differences in them, then oxygen coping machinery in low oxygen condition might be different. So now then, does this EGLN1 become a molecular correlate of Tridosha? So Tridosha has properties that it is pervasive, it works across the system, it works right from the time of birth, it can govern many conditions. So what we actually find is that many of the features of Tridosha are kind of satisfied with this one. But what is very interesting is that hypoxia is a very important axis. If you perturb hypoxia, you can actually EGLN1 variability. People have shown that cartilage, wound, brain tumor, renal anemia, cardiovascular disease, myocardial treatment of inflammatory disease, everywhere EGLN1 is a potential target. Either you need to increase hypoxia, like in pre-ischemic conditioning, or you need to reduce that, for example, in cancerous condition. One of my colleagues, Dr. Anurag Agarwal, he looked at the same axis in asthma in mouse model, and he could actually show that modulating this axis actually exacerbated asthma by causing more and more inflammation, such that it became steroid resistant. Now, having this model, it was very interesting. Now we have a pith cuff axis of EGLN1, a mouse with asthma, which has inflammation consequence. If you modulate that, we asked the Ayurveda doctor that if we actually take a drug which is used to treat pith cuff perturbation in asthma, how does it work in this mouse model? So this is the experiment which was done by a modern doctor and an Ayurveda doctor, Bhavna and Anurag Agarwal. And it was shown that actually, if you do this, it actually reverts. So Adatoda Vasika was a drug, Vasa, which all of you know very well. If it was taken, it actually brought down airway inflammation. It brought down all the markers which are indicative of steroid resistant. Not only that, it actually restored mitochondrial shape and function. So now you can see that using Trisutra concept, you have a difference in pith cuff you identify from phenotype. That becomes relevant in a disease where hypoxia is relevant. Take that axis and perturb it, you find in a model asthma can be modulated. Now you take a drug of Ayurveda which works on asthma and brings back pith cuff, you find that it actually alleviates hypoxic signatures. It actually, we showed by doing the lung transcriptome that hypoxia axis was alleviated, and it was a prolyl HIF1 alpha inhibitor. But what was very interesting is that when COVID came, so since it was useful in hypoxia, the student of ours tested it in sepsis and other models of hypoxia. He found it is relevant. And then when COVID came, in the preclinical model, this drug was active. So this actually suggested that during COVID time, Ayurveda doctors recommended Vasa as one of the drugs to be added in their recommendation. So, and we also realized hypoxia was a major cause of concern. So if one knows that Adatoda Vasika has this kind of a property, even modern medicine people should acknowledge that, okay, in an integrative medicine setting, this can be actually useful in a particular kind of a case. And then hypoxia even became more relevant that now we have found that hypoxia, inter-individual variability in hypoxia governs responsiveness to HAPE. We also had a paper where we showed that it could also govern thrombotic outcome, that is bleeding or thrombosis, who will get it in hypoxia, was governed by pith cuff differences at the genetic level. So these all become relevant, that in COVID time, who are the people who might actually have bleeding outcome, who might have thrombotic outcome, who might have hypoxia problem. Those are, supposing you were to do prakriti of people with different kinds of COVID outcome, would they have differences? Testable. I don't know whether it was tested by Ayurveda community, but this is what our result told. This was done by a very big team at IGIB, all this work. Bhavna and I co-led, and there were lots of doctors. And but how do we move ahead, right? So this is like 2 years back. I moved to Jodhpur, and this is something which now we have built a framework where you can develop a working hypothesis, you can test the working hypothesis, you can validate it, and you can use it for repurposing in Trisutra Tridosha framework. What we had also done was, during this time, we had worked in the lab, but we wanted to kind of see whether we can develop more objectivity. So there is where we worked with Sanjay Juvekar and his team in Vadu, and we looked at nearly 10,000 households, looked at their prakriti, developed a questionnaire, and then in that place, we actually looked at more genes for pharmacogenomic relevance. We found that there were variations in them which were important for many responsiveness outcome. And this I'm giving you a hint because in your breakout sessions, you will have to start thinking about whatever problems you are facing with. Very recently, we also showed that if you take extreme prakriti types who have most predominant Vata, Pitta and Kapha, and you do exome sequencing in them, that is, you look at their genetic variation, do you see elevated frequency of variations which are associated with different risk for different diseases? And this we did in both two cohorts, and we got a large number of variations intersecting with variations which have been associated in many worldwide genome-wide association studies, which tells us that we can actually identify such kind of informative variation and start to assign risk scores for different diseases. And even if we use prakriti as an additional way to non-invasively phenotype people, we might be able to kind of attribute certain kinds of risk to certain diseases. So for example, on the left side, these are all different kinds of diseases. On the right side, there are all the variations which have been reported, and these are all different kinds of things. So you can see multi-system attributes have genetic variations which can be used for assessing or for risk stratification. We've also used machine learning methods. That is, supposing we take Ayurveda features and we remove the Ayurveda labels and ask the machines to cluster the people on the basis of Ayurveda phenotypic attributes, which we've captured. These are all attributes of anatomy, physiology, and all. Do people bin separately? And the answer was yes. We did unsupervised machine learning, and we found that they could be separated. And then our students developed supervised clustering method, and they used it to a set of people who had been faculty has been assessed using the same questionnaire, and we try to identify would we be able to assess who are Vata, Pitta and Kapha. So this actually tells that if such kind of machine learning approaches can recapitulate what Ayurveda doctors have been assessing, in that case, there is a possibility to develop methodologies which become a tool for assisting Ayurveda doctor in clinical settings and achieving more objectivity. So we've been able to do that. We've also looked at gut microbiome in people who are of different prakriti types in Vadu cohort only, and we find that extreme prakriti individuals have endophenotypes or some microbial signatures, which are also associated with predisposition to different diseases. You cannot classify people just on the basis of microbiome, but if you classify Prakriti on the Prakriti and look at their microbiome, there are differences in them. So that actually tells that we could do that. And this is becoming very just now presented. Recently, Bhavna with Dr. Deepak in AIMS, so again, a physiology and a Ayurveda doctor, we could show that heart rate variability, baseline heart rate variability does not differ, but if you give a stress like head up tilt table test, then the response was between Vat and Pitta, Vat and Kapha, which tells that Vat actually responds much more, their parasympathetic component is much more than the Kapha. So this actually suggests that given a stress, the responsiveness to autonomic functions might differ between Prakriti type, which is also known from the physiological questionnaire which Ayurveda people have been asking. So that way, a large number of readouts from various levels suggests that Ayurveda Prakriti could be a one very important tool for integration in risk stratification, early intervention. This is just one last part I will just deal with. So this was like this we started not on COVID, but before COVID, and it was prompted by a question from my lab itself, that supposing we have this cause, feature, and disease, then would you be able to use Ayurveda drug to also identify links between the causes and the features? So why I use this is what one Ayurveda doctor treats multiple diseases, and sometimes he uses the same drug to treat multiple diseases. How does he do that? For modern people, it sounds like, how is this possible? We have specialties for so many diseases, and how can one do that? My question was that if that was so, is there a crosstalk between the pathways to these diseases, which is coming out in all the perspective, or are there, because they work with polypharmacology, multiple molecules are there, so some molecules go and treat this disease, some molecules go and treat the other disease. So we took a drug called Cisamplas parera. It's called Patha by Ayurveda doctors. It's used in Africa population also, but in Ayurveda, it's used for treating hormonal disorder and fibroids, very high fever, febrifuge, and it's also used for snake bite. Very interestingly, whichever populations are very malaria endemic, this also drug is this medicinal plant is also present. So I asked a question that is there a possibility that is there a link between estrogen and antiviral inhibition, or is this drug working by two different methods? So we developed a platform based on a platform which is being developed by MIT. It's called Connectivity Map. So what is done in this here is that if there is a drug or any patient samples, we can treat the drug on a cell line and get a expression signature, that is gene going up, going down, like that. Or even from a patient sample, you can actually get such a signature. And this signature you can query in this database, which is made by MIT Broad, where nearly millions of such compounds have been treated in such a kind of a platform to get the signatures. So now what you can do is computationally, you can connect or you can correlate these signatures to other drug signatures which are there. And if this matches to certain kinds of drugs and you know the classes of the drugs, then you might be able to kind of say that our drug matches what kind of signatures. So it's basically pattern matching. Your drug matches the pattern of existing pattern, then you infer the signature. That's what is used for repurposing. So we did for Cisamplas parera the same thing, and we actually ended up finding a large number of signatures which are linked to protein synthesis inhibition drugs, which are protein synthesis inhibitor and used for dengue. But it has side effects, so they are not used, but those signatures were there. So we went to a dengue expert who works on cell lines and treats with dengue, and we gave this drug and we found that it inhibited. But that was part of the story where is the estrogen link. So we went further and looked at this array, because now we had a knowledge of Ayurveda that it works on estrogen. We found that there was a cholesterol synthesis pathway which had gone down, and estrogen receptor had also gone down in expression. So we took the molecule drug, bit on estrogen receptor, and there were 38 molecules which actually could bind to the same region where tamoxifen, that is an anti-cancerous drug, binds, and a beta estradiol also binds. So then we found that it also binds to a protein synthesis inhibitor protein. So what we did was we asked the collaborator to treat the same dengue lysate where this drug was used with again an estrogen antagonist and see what happens to the drug action. We found that actually the drug action was modulated when this was done. So this was the first time we could actually show that there's an estrogenic link to this drug, and that is why maybe Ayurveda doctors use it for this and the other. But then what was interesting was that COVID we then and this was there was a link. So we had done lots of validation for estrogen axis and all. But then COVID came, and somebody had actually done an expression on a COVID patient. So we took that expression also and put it through this box. And we found that CIPA, what it was taking up was actually, meaning the drug, there was an opposite signature of the COVID with the CIPA, or the drug which we tested. So then and also the estrogen axis was also there. So we went to CCMB and asked them to see whether if we give this whole compound or we give single molecules from this compound, is it able to reduce the titer of SARS-CoV-2 in an infection model? And we found actually is a extract, it actually brings it back down to 90%, and many of the single molecules together are, meaning at single level, they are able to bring 50, 60, 70, 80, but not the complete as we see for the whole extract. So this was the time we could actually show that if Ayurveda doctors were to use Sesame plus Pereira in COVID time, then maybe that might also have a effect. During COVID time, Ayurveda doctor did not recommend Patha. So went and asked the doctors personally. They said that it is there. Descriptions are also there. They got out a shloka. But during those times, there was to be a plant which was available in all parts of the country, which was easily made. Ayush 64 was already in place. So now if we give this evidence, Ayurveda people say that if SARS-CoV-2 kind of situation comes, then Sesame plus Pereira is also something which they might be able to use. So now you can appreciate that integrative medicine might actually, by this kind of an approach, might make it interoperable, complement, synergize, and it can actually give more options to a patient in times such as like the COVID time. So I'll try to kind of give you a scenario. I was telling about the other people, but this is my personalized health journey. And now, because I have delved into Ayurgenomics, I'm not an Ayurveda person, but I have gone through all these kinds of things in my whole lifetime. You all might be able to kind of assess that my Prakriti has been invariant. Only thing is manifestation of my Prakriti has happened at different times. So for example, early age, severe sweating, acne, dental problem. In the middle age, AVM bleed, moderate obesity, fibroids, again dental problem. And now in the desert environment, which induces Vat, age which induces Vat, I have aging problems, the dental continues. So from all these things, I feel that I might have been Pith here, and now Vat. So I might be Vat Pith, but Vat would be much lower, but Pith is much more aggravated. And I need to keep the Pith in check. Maybe my diabetes might be cured just by managing the Pith and Vat in a proper proportion. But this has been my journey, but I'm just saying that if we are able to fit this jigsaw puzzle by a combination of methods, we might be good. So in IIT, now we are just doing this. We are kind of integrating it with data science, engineering, and multi-omics. to be able to build ontologies and to be able to use it for large scale applications. There's a unique research ecosystem there, which is very, very happening. And we are working on diagnostic, therapeutics, validation, capacity building, all these areas, primarily for Prakriti, Vikriti, Panchkarma, integrative medicine setting, and also kind of globalization. Basically, if we can actually create a virtual health guidance for a person in a participatory format, adding a layer to P4 as a P5 as an Ayurveda Prakriti component. So I'll not get into all these, but we are doing many, many kind of activities where we are doing Ayur Thinkathon, taking Ayurveda doctors, exposing them to various kinds of technologies and asking them to think a shloka and connect to technologies and make a presentation of a project. So there's a big group with us. Professor Chaudhary, who is our director, he works in the area of computer vision. He's an AI person. There are sensor people, there are computer people, computer science people. There are industry partners, electrical engineering, chemistry. So, and we are building the team because as you realize, this cannot be done in one place. It needs to be validated in a large number of settings, and many people have to come on board. And we've now got this Ayurtech Center of Excellence established with the support from Ministry of Ayush. And Rajasthan Ayurveda University is very 1 kilometer away from us. So there are lots of charged MD doctors who now want to do PhD with us. So we are developing unique programs. So there's a whole lot of people who are Ayurveda doctors who are working with us. And AIMS Jodhpur is one place where we have initiated a integrative medicine clinic in January, where doctors are sitting across the table and trying to formulate how to go about giving a joint recommendation. We are not clear. The doctors are not clear because despite 10 years of coexistence, this is the first time they are sitting and discussing together. Otherwise, it was referral without kind of cross-talking. So this is it. Thank you so much for patient listening. And I think we can do many, many interesting things if we actually get into the spirit of collaboration and start working together. Thanks. I think I overshot my time.
Thank you, Mitali ma'am, for such insightful lectures. And I am here to introduce we are moving into the next session. And so this session involves each one of you, each one in the audience. We all were, I'm including myself in the students because I'm still a student of Ayurgenomics here. So we all were handpicked by our advisors, by the head of our departments to be here. So let's make something worth of it. And this is where this workshop differs from all the other workshops that I have attended personally also. And I'm sure you all have the same experience. So next session is we'll have like 4 small capsule talks. And after that, there'll be breakout session. So in the breakout session, we'll make 4 groups. Each group, if you see, there will be like 4 topics that we are going to discuss: inherited genetic disorders, which will focus on sickle cell anemia; non-communicable diseases, which will have a focus on diabetes; oncology; and health and wellness. And we all will be in 4 groups. And this is where we will benefit. We will get to interact with the speakers one-on-one. So that will be the time where each student, I want all the ideas, crazy or not, they should be on the table. Something good will come out of it, I am sure. And with the guidance of all the honorable mentors that we have here, I think we are going to get some nice ideas, and we're going to be on a path to do some great research. As VC ma'am had said earlier, we are in a workshop and we all have to work. So next 4 session, please listen very carefully. And again, I want to introduce the first speaker, Dr. Gambhir, Dr. Prakash Gambhir. He actually, he is the one who put the foundation for the GIDN department at Regional Center of Pune. I consider him to be a mentor. I run to him for advice anytime I face issues. He is presently the Chief Medical Scientist in LifeCell and Phytomed. He is the Director of Birthright Genetic Clinic, Pune. He is the President of the Genetic Chapter of Indian Academy of Pediatrics. He has more than 40 publications in national, international journals. His research interests include newborn screening, fetal dysmorphology, lysosomal storage disorders, prenatal screening, and basically education of genetics. So he is a huge resource for us. And after the talk, during the breakout session, if he's available, you can ask him questions. I would request sir to please come on the stage.
At the outset, let me thank Honorable Vice Chancellor, MUHS, Dr. Kanikar Madam, and the faculty at NCCS, Honorable and Mansi for the presentation. Dr. Vitali Mukherjee and Sukhya have given such a nice talk. In fact, Mansi said that she is a student, I'm also a student.
genetics है. So I was wondering कि I'm a physician, what do I know about genetics? And honestly, I've not worked in genetics related to diabetes at all. So he said, okay, sir, you tell us the problems which are there. So that made me a little comfortable, ma'am. So I'm here like a schoolboy with lot of problems here. I'm showing you what are the issues about diabetes. And I hope at the end of this talk, we'll get some answers. Very honestly, I did study about genetics of diabetes in last 3-4 days when sir called me, madam. It was thoroughly taxing, but I have got some slides which I'll show if time permits, because I've been warned that I have to stick to 15 minutes. So I'll quickly tell you what are the issues in diabetes in the world across and around country. And madam, I must say I'm fascinated to do something in Ayurveda already. That's what I have planned to talk about. So the question is, next. Okay. So what is bothering us? Our problem is that India has been referred to as the diabetes capital of the world, which I don't think is a very good title for any Indian to follow. We've been labeled as the diabetes capital of the world, and as it accounts for around 17% of the total number of diabetes patients in the world. And very large number, around 80 million people with diabetes are there in our country, and the number is expected to increase to 135 million by 2045. That is very alarming. And since my undergraduate, postgraduate days, and as a teacher, I've been treating diabetes, and I've seen that it is ever increasing. So we all know that it is the largest global health emergency. It ranks among the 10 leading causes of mortality together with cardiovascular disease. We know, like whatever, I'm a physician and I see patients in the ward, most of them are diabetic, hypertensive, and ischemic heart diseases. And these three friends are the worst enemies of mankind. According to the WHO, non-communicable diseases accounted for 74% of death in 2019, of which diabetes resulted in 1.6 million, which is a very large number, and it became the 9th leading cause of death globally. And by 2035, we expect to have around 6 million people who may die of diabetes. And type 2 diabetes constitutes 90% of all cases of diabetes, which was earlier considered to be a disease of affluent Western countries. But it is not so anymore. Globally, it has been shown that it affects the lower socioeconomic status as well, and younger population also. It has reached an epidemic proportion in many developing economies such as China and India. And of course, according to the WHO, the prevalence is growing most rapidly in the low and middle income countries. And the reason is there is a rapid socioeconomic change with urbanization and industrialization, which is the most important factor. I'll be briefly touching on to the issues and challenges, which will highlight what, as physicians, we have the difficulty in treating these cases. The global burden is variable. It varies from country to country. I will not elaborate on that. The International Diabetes Federation says 8.8% of the adult population have diabetes all over the world. Men have slightly higher rates. I'll quickly go to the Indian perspective. The top 3 countries with the highest number of individuals with diabetes are China, around 116 million, India, 77 million, and the United States of America, 31. So we are number 2, but then we are projected to be number 1 by 2045. This trend is going to continue by the period of 30 to 45, and China will still lead, and India is going to be the second, and we will have a continued burden of diabetes in future also. And remember that the growth in population and the aging factor is responsible for the increasing burden. These are little busy slides. I didn't cut them short because I wanted to highlight all the various problems, but since the time is less, I'm going to quickly read through. The global burden of disease study conducted in 195 countries and territories. It gave a detailed overview. It reported that low and middle income regions had higher burden of diabetes, while the high income regions had lower burden of diabetes. This study reported that people with incident diabetes increased from 11.3 to 22.9 million between 19 to 17, and they are still on an increase. And the increase was a very phenomenal 102% of the existing number within the 17-year period. And the number of prevalent diabetes increased from 211 to 476, which was an increase of 129%. It only highlights that the problem is increasing, will keep on increasing, will ever increase, unless, of course, Madam gives us some breakthrough to control this in future. The problem of undiagnosed diabetes, we are not aware. In a study, I'll just tell you that there are many studies which have been shown, but 59.7% of the people are actually not aware that they have diabetes. And the same is true all over the world in varying numbers. Now, this is what is the graph which shows that the proportion of number of individuals with undiagnosed diabetes, according to IDF, it differs. And we are here, wherein in our country itself, the unawareness about their own diabetic status is very significant. Well, let's quickly talk about what is happening in our own country. Diabetes has increased steadily in the last 3 decades. Remember that there is an epidemiological transition. Initially, there was mortality from communicable diseases, maternal, neonatal, and nutritional disease. But now that has changed. Now there are more deaths because of non-communicable diseases, and diabetes accounts for a major amount. The prevalence of diabetes in India has increased from 7.1% in 2009 to 8.9% in 2019. And at present, we have around 25 million people to have impaired glucose tolerance. India ranks second after China, that we've already talked about. And of this 12.5 million aged or 65 years, which is estimated to be 27 million by 2045. So the elderly are going to be affected more in future. Now, this is the graph that shows an increasing prevalence of the diabetes during the last 10 years. I'll just skip this. We'll just focus on the pre-diabetes part. There are a lot of patients who have pre-diabetes. Their sugars, fasting sugars are impaired, though not in the diabetic range. They're not aware. And the progression from pre-diabetes to diabetes is very important. These are the population we have to target and then accordingly treat and manage. The prevalence of pre-diabetes ranged in our country from 5.8 to 14.7 in rural India and urban area respectively. And the prevalence of pre-diabetes exceeds that of diabetes. That is very important. Diagnosed on treatment is one thing, but pre-diabetics, who are the major group, needs to be focused and looked into. This is the state-wise prevalence of diabetes and pre-diabetes, wherein you can see that certain states like Chandigarh and the South Indian states like Tamil Nadu, Karnataka and Maharashtra, they have large number of pre-diabetes and diabetic patients. Progression from euglycemia to pre-diabetes and pre-diabetes to diabetes. This is another important thing. There are a lot of people, there were studies which showed that the progression of people from euglycemia or normal diabetic status to pre-diabetes was faster, and of course from pre-diabetes to diabetes or more. So actually these people with pre-diabetics have to be followed very religiously, and a lot of work has been done down south in Chennai by Professor V. Mohan about the patients of diabetes, and he reported that the conversion rate to diabetes was 19.4% among those with normal glucose tolerance and 58.9% among those with pre-diabetes. So the progression from normal to diabetes was lower compared to pre-diabetes to diabetes, and therefore this again I emphasize should be the focus of our care. The similar thing was shown in Kerala and down in Pondicherry. This is the burden of diabetes and pre-diabetes in India, which I have already showed in the previous slides. So we have the impaired glucose tolerance estimates, which was 25.25 in 2019 and is expected to be 35.7 million by 2045. And that is where we are being, we are heading to. And the undiagnosed diabetes estimates are, as I told you, around 57% people, they are not aware of their diabetic status or they are pre-diabetic already. This is very important. Why are we so worried about diabetes? It is an important cause of morbidity and mortality. A patient rise of hypertension has underlying diabetes. A patient rise of heart disease has underlying diabetes. A patient has a massive amputation, the underlying disease is diabetes. A patient loses an eye, there's underlying diabetes. So it is an important cause of mortality and morbidity all over the world. And we all are very well aware of that. And remember that the rural areas and urban areas are equally affected by this condition. And here, this slide shows what I was talking about, that in 1990, the deaths due to communicable diseases, infections was much more compared to the non-communicable diseases and injuries. But now here, non-communicable diseases have taken over and diabetes accounts for a major chunk of the cause of death in our Indian setup. We all know that diabetic retinopathy, the most common cause of blindness after cataract is diabetic retinopathy. It is an important marker of the severity of the disease. And of course, in a study, they showed that 1/5 of the people with diabetes had diabetic retinopathy, and a lot of them had blindness later. We are aware that there are microvascular complications like ischemic heart disease, strokes, other things. And this study, the study which was done in China showed the various percentage, which range from 33% presented with microvascular complications, wherein neuropathy, nephropathy, and ocular lesions accounted for around 30%, 6.8%, 17.8%, etc. So basically, I'm trying to highlight that the complications are very severe, and in large number of our diabetic patients are suffering from this. There's a very high mortality. I don't think I have to overemphasize the fact that patients of diabetes die in much more numbers compared to non-diabetics by the virtue of the microvascular complications like ischemic heart disease, infarct, and brain strokes. Well, what are the risk factors? There are modifiable risk factors. Modifiable risk factors like sedentary lifestyle, obesity, unhealthy diet and habits, stress and depression, altered environment, environmental pollution, and inadequate sleep are the modifiable risk factors. We cannot cure disease. I think all of us know that whether it is type 1 diabetes, type 2 diabetes, or the intermediate diabetic syndromes, all these are genetic immune diseases. Okay, so we cannot do anything much for them, but can we try to modify some factors? Yes. And modification of these factors like obesity, diet will definitely help to reduce the severity and progression. The non-modifiable risk factors, where I think today's session is mainly on genomics. So genetic predisposition, family history, race, ethnicity, can we modify? Madam, I always had a lot of questions in my mind that why not all people get diabetes, number one. Two, why is that in a family? Only few siblings would get diabetes, the others would be spared. Why, if only the mother was a diabetic, there would be less people of diabetes in the family. If the father was alone diabetic, again less. But when both were diabetics, the numbers are more. I think, madam, the answer lies in the genetic parts and the genomics of it. You didn't talk much about diabetes during your talk. So maybe during the second half of the day, we get to hear something about if there's anything been done about diabetes. So these risk factors are, so what we have are these problems. Sir, I want to specify these problems because what are the challenges we face? We do not have a strong national partnership, multi-sectorial actions. We do have lack of ability of surveillance. There is very low disease awareness among public and lack of basic prevention and management diabetes facilities. Disproportionate fund allocation for diabetes, difficulties engaging the industry and private sector, limited human resources, and inadequate community mobilization. And these are our problems. Actually, when I as a physician treat diabetes and I see the large number of cases of diabetes, this is our issue. And what can we do to prevent? Yes, we can reduce the number of these cases of diabetes by addressing each risk factor, either alone or collectively. So lifestyle modification, early detection, a very robust surveillance, and then national food policies. We have to target on this kind of food our people eat and can be provided with quality food. I won't go into the details of that. We have to reduce harmful behaviors, prevention policies, and all this will, and of course, to reduce the cost of essential drugs. I'm in a government hospital. I've seen my patients not getting drugs for 1 month, 2 months, because they're so poor, they cannot even buy drugs worth 50 rupees. And once they miss drugs, everything goes off track. So I think this is one thing which we'll have to do. Ma'am, I'm very tempted to show you that I have exerted to talk about the genetics also, but I'll just rush through this. We all know that the genetics it plays an important role in diabetes. There are HLA genes, non-HLA genes. Obesity and obesity-related genes are important, protective as well as harmful. It's a spectrum of disease, lot of genes, and there are a lot of immune diabetes, especially MOD. Lot of work has been done, and these are the various type 2 diabetes is been told to be a genetic disease with modified risk factors which we can modify, but at the base it is a genetic disease, and a lot of studies have been done. I mentioned about these GWAS studies, which are actually trying to study a lot of genes and tell us about the pathogenesis. Probably the idea is to find out the gene that can be modified, give these patients gene therapy, and either reduce the incidence of diabetes or to prevent the complications of diabetes. I'm sure the kind of work ma'am is doing, the day will dawn soon. Thank you so much.
Thank you, sir, for all the questions on diabetes. There is a project going on with MUHS. In the breakout session, we'll tell you the details about it. So I would request Major General Dr. Jahaz, sir, to please come on stage and felicitate Prasad, sir. Yes.
But I leave people with a food for thought of when he's given the problem statement, but in diabetes, I mean, can you look at give your answers to the Ayurveda? I'll be joining. I'll be joining in online and continuing to be part of it to travel. But I'll leave you food for thought because diabetes is not only statistics, but we've heard since morning, and he's given us the huge problem which is there. But we've also heard that he said, I don't know why. Why some people don't get into a situation? Why people behave differently? Why some people go into a renal manifestation and some the eyes is. And some, in spite of a good HPV and ACE target organ involvement, some don't. So I leave you with that food for thought.
Thank you, ma'am.
Good afternoon. So after listening to all brainstorming lectures and inspirational talks from eminent speakers, now I've been given the privilege to announce Dr. Sharmila Bapat, whose talk is going to be very inspiring and close to my heart because I'm also a cancer researcher. So here there is some of the list, long list, but I will just highlight some of them. She is an independent researcher in NCCS. And to point out some special academic appointments, she has been from 2016 to 18 member American Association of Cancer Research, that is AACR, on the Advisory Committee of India, and external member Board of Studies of HBNI University, from where also I stand as a professor from HBNI. There are several honors and awards like R.M. Tiwari Research Oration Award, and Outreach Lecture Fund Award of United States International Education Fund. And last but not the least, in 2023, recently, she has been Sri Ramnik Lal Kinnariwala Cancer Research Award, Gujarat Cancer Society. And I'm proud to say last year I was the recipient of this award. And it's a very nice to have Dr. Sharmila Bapat receiving the same award. And she has over 45 publications, and majoritarily I hope they are in ovarian cancer, which I've gone through that. And her research areas have been especially in cancer stem cells, epithelial, that is EMT transition, gene regulation, tumor heterogeneity, biomarkers, clinical stratification of molecular subtypes in ovarian cancer, and publications in peer-reviewed journals like Cancer Research, Oncogene, Semin Cancer Biology, and so on. Like the big list is very big, and she has around book chapters, 14 book chapters, and nearly 4 patents, which she has worked upon. And I invite Dr. Sharmila Bapat for her excellent talk. Thank you so much. Please.
So, ma'am, we'll have a 2 minutes break. I request all speakers to please proceed outside because VC ma'am is leaving. So we'll have a photograph session with VC madam.
I'll just put it in.
I'll do it. Thank you so much.
मैम दिस मैम अपन दिस करें कॉपी पेस्ट करो ठीक है चलो
Welcome back after nice lunch, and I am seeing everybody, and everybody looks awake, which is a very good sign. Let's keep it that way for another half an hour, and then break also for the breakout session. So, we had introduced Sharmila Bapat madam, and now I request her to start with the presentation. Also, I just want to tell all the speakers जो पहले नहीं कर पाए, Barda sir and Bapat madam, that VC madam has the link, and she would be watching from her car because she's traveling right now. So thank you.
So at the outset, I usually thank my organizers, but today I must thank all of you who have actually come back after such a lovely lunch. So thank you all for being here. And of course, now I have to thank the organizers. When last week, when Dr. Saurav Sen called up to ask me whether I would give a talk, I said, "What on what?" He said, "Personalized oncogenomics." I said, "Okay, that's fine. That's just up my street, so that I can do easily." What I didn't know was that there was going to be a crosstalk with Ayurvedic Ayurgenomics. And then I realized what the task was set up for me to do. So, but I'm really grateful for this opportunity because it's given me some impetus to study something that I would have otherwise not really looked into. Although I didn't know of Mitali's work earlier, I've heard her so often, and it's always impressive. So, my lab at the NCCS works in the area of ovarian cancer research. Over the last 22 to 23 years, we've been working on a histopathological subtype known as high-grade serous ovarian cancer, which is responsible for the highest mortalities amongst all the gynecological cancers. One of the reasons for this high mortality is that there is a very low frequency or success of early detection of this cancer. And as a result of which it is usually detected when it's already metastasized and has peritoneal ascites that is formed, which is already a high level of metastasis. So very often patients who are treated early on immediate detection of the disease, they do respond. In fact, 70% of patients respond, which is surprising, although because it's at a metastatic state. However, most of them do not get into remission, and about 60 to 70 of these actually come back with a recurrent disease, which is highly aggressive and is associated with a lot of mortality. So we started looking at this as a challenge because there were no researchers working on this particular subtype or ovarian cancer at all in 20 years ago. And we thought that we would try to do something that was really challenging enough. Over the years, we've done a lot of course, as being a cell biology lab, we have been doing a lot of experimentation. We have what is now very broadly referred to as siloed data. But for me, they are all important experiments, important identifications about cellular behavior that, give you very tiny clues. And when you try to fit it into the disease perspective, And that is very important to do because you cannot identify these cues when you look at the disease at a whole. So I think it is very important to have this crosstalk between the silo data or the experimental data, which may be done in a cell line, in a few cell lines, or with a few primary samples, and then look at it across to the disease perspective on a very broad overview perspective. The best way to do this is look at the omics data. You have the epigenetics data, you have the genetics, the transcriptomics, proteomics, metabolomics data, which give you a very rounded and a overall picture of the entire disease. Now, this is what essentially that we've been doing in the lab with all the data that is generated. We try to put it in the perspective of the disease and try to understand how we can maybe help understand the disease and eventually the patient in their recovery. What we hope to move towards is always smart or informed data. We've been doing a systems networks analysis, although we have not got into machine learning or computer or cognitive computing, as it is called. And of course, the whole sum of it, the big picture, as I say, is going to be evaluated by its relevance and applications in healthcare. So coming to the topic that was given to me, over the last several years, in decades actually, I would go on to say, people have been realizing, especially in oncology, wherein you have very large or broad spectrum chemotherapeutic drugs that are available. They are often used across different cancers. The same drugs are used across different cancers, and all of them have different outcomes in different populations with a different ethnic background, and so many differences that are actually ingrained in human life across the world. So this has actually led to the realization that each patient actually is genetically different. So the tumors are also likely to be different, and that necessitates that the therapy that is developed for a patient in order to be effective must take into account all these differences in order to be really effective. So this means that one has to, what is a term that is very often used is find the right medicine for the right patient, or you match patients with drugs. Now, this is very easily said than done because it involves a lot of parameters to be studied. You have a lot of, currently you have a lot of clinical research efforts that are going on to see or which incorporate data from different facets, including the clinical data, the patient biochemistry, and so on and so forth, in order to find out what the patient is likely to respond to. And most of these personalized approaches, they are found to be able to contribute to an overall improvement in the outcome, in patient outcomes in terms of overall survivals. However, cancer still remains a disease that is dreaded because nothing is certain as yet. And it's this uncertainty that most of us always try to address. I was fortunate to meet a person, a woman called as Laurel Pratt in 2016. At that time, she was a 19-year survivor of high-grade serous ovarian cancer. And to my mind, I just talked to her last year. Today, she is a 23-year survivor of high-grade serous ovarian cancer. And I have never heard of a patient with HGSC who has survived that long. So it's very encouraging to know that there can be such long-term positive outcomes. And I think that is the aim with which my group works in order to make this possible for a large number of patients, for a larger number of patients, rather than the single lone one that we know. So what exactly is the concept of personalized oncomedicine? Primarily, one studies the family history. It's very well known that there are germline mutations that are inherited, that are passed from one generation to the next, and that indicate an increased risk, or it's a predisposing factor for acquiring specific cancers. I think the most cited one is of the BRCA1, BRCA2 mutations, which are present in several, it's also associated with the Lee-Fraumeni syndrome. And it's a predisposing mutation for breast and ovarian cancer. Now, this earlier was thought to be a very, as a very threatening kind of a diagnosis. But the developments over the last several decades have resulted in the outlining of PARP inhibitors, which are known to be effective in a large fraction of these patients. Now, surprisingly, although the BRCA1, BRCA2 mutations or the gene itself was identified in the context of breast cancer, the responders have been more in ovarian cancer than in breast cancer. And this indicates that there is possibly a tissue specific context to the efficacy of drugs also. And there are people who are trying to work on this, trying to find out, even in the absence of BRCA1, BRCA2 mutations, some patients actually respond to PARP inhibitors. That has driven a field called as identification, that tries to find signatures such as a BRCA-ness type of a signature, which will be useful in identifying patients that are likely to respond to these PARP inhibitors. Now, over the last decade, whole genome profiling and deep sequencing or even transcriptomic sequencing has identified tumor-associated mutations, and some of these mutations are actually known to be druggable. The best cited instance is in lung cancer, wherein we have a knowledge of several of these non-synonymous mutations, which are usually point mutations that result in the change of one amino acid, or the codon, that is the new codon actually codes for a new amino acid rather than the previous canonical codon sequence, or the trinucleotide sequence that was present in the gene. So these have been known to be responsive, and now there is an entire line of treatment that is based on targeting these, and hence they've been termed as druggable mutations. And there's a big search, there's a big effort on the way to identify druggable mutations, additional druggable mutations in lung cancer and across all the cancer types. So this is a huge effort in genomics that is really yielding some indications about where therapy could be developed further. Now, this has also led to the formulation of hot mutation panels or mutational panels, which are useful across cancers. Now, it's surprising also to find that the same mutation may be involved in different cancers. For example, I mentioned BRCA1 mutations, wherein PARP inhibitors have been shown to be useful in ovarian cancer patients. Surprisingly, even in pancreatic cancer patients, there are some recent reports. Pancreatic cancer is known to be just as aggressive and fatal as ovarian cancer. So it's quite surprising, and it's actually quite an enthusing finding to know that a fraction of patients who have this kind of a BRCA-ness signature would respond to PARP inhibitors. So you can have again the same type of a genomic change, which will lead to a new therapy. So I've been told that I have only 2 minutes to go. So let me rush. I don't know what to talk about then, but let me think. I will share with you what I thought. These are just approaches in personalized medicines that are thought of. There are obviously a lot of limitations to personalized medicines, apart from just the cost, which may actually be reduced as the technologies improve, but there are a lot of issues. And I thought that yesterday, actually, I spent a good amount of time trying to think where Ayurveda or Ayurgenomics can actually have an overlap with some of the features of genomics, cancer genomics, and where it will be beneficial to step in. So these are some of the things that I thought of. I'll just refer to a study that was done in my lab almost 10 years ago, wherein we although I talked of being a researcher in the field of high-grade serum ovarian cancer, we had soon realized that this is a heterogeneous disease. And using a lot of statistical methods, we had used the transcriptomics data from the Cancer Genome Atlas to stratify these or group them into 3 types of groups, each of which have a very distinctive molecular signature. Now, this class 1, we just named them class 1, class 2, and class 3 patients. As you can see, class 1 and class 2 patients had some modules. Now, each of these tiny square in this module represents a single gene. So we had a lot of genes that correlated positively with each other within the class and negatively with several other tumors, which segregated out as a separate class. We had another class which seemed to be presenting with mixed features. And this is what we actually was confusing. So initially we started looking at the biological behavior of this. And in this, what we found is that the class 1 represents tumors which have a strongly epithelial phenotype. They undergo aberrant differentiation, genetic instability. The cells actually proliferate very fast, and they have a very aberrant tissue homeostasis patterns. So since morning when I was listening to all the inputs of Ayurveda, I was wondering whether it would be able to actually stratify this into one particular Ayurvedic class, because the other class is just the opposite way. It is highly invasive. It undergoes an epithelial to mesenchymal transition, as a result of which the cells are mostly mesenchymal in nature. They exhibit a lot of phenotypic plasticity. They are actually quite robustly maintained. Genetic instability is not a feature that is associated with these, and they have a huge amount of crosstalk, which is probably triggered off by the extracellular matrix with the immune cells in the microenvironment, and they have something called as the interferon gamma triggered pathways. So I have been thinking, okay, this is something that we had taken ahead and validated in clinical samples. It was not purely a bioinformatics based stratification, and with the help of several collaborating histopathologists, I think I have them here, in AFMC, KEM, in Lice Budrani Hospital, TMC, we have actually validated the prevalence of these 3 different types of tumor subclasses amongst patients. And we were able to identify this using a panel of 6 biomarkers. We tested several, but out of these 6 seem to be able to cover most of the samples which have been stratified as HGSC. So today what we do know is that we have these 3 subclasses. Each of one, because of their distinctive molecular patterns, is likely to respond different to different drugs. What we also did is that we developed a monoclonal antibody, and what worked was a monoclonal antibody against the mesenchymal subtype. This is something that we termed as MEG150. The monoclonal antibody is a cytotoxic antibody, and it recognizes a molecule called as annexin A2, which is present on the surface of tumor cells. We have combined, as you can see, these are the preclinical explorations, wherein you can see a reduced tumor size on treatment with just the monoclonal antibody again by itself. However, one knows that this residual tumor has the potential to actually regrow back and give rise to a more aggressive or an enriched phenotype. So what we decided to test out is to look at the expression of or the efficacy of this monoclonal antibody in a combination with 3 different types of drugs. One is gemcitabine, which is already in the clinic for ovarian cancer and which targets the rapidly proliferating cells. One was a HDAC inhibitor, or a deacetylase actually, 5-AZA-DC, which desuppresses the promoter methylation of the target gene of the monoclonal antibody. So as a result of which the efficacy of this monoclonal antibody is increased, as you have a larger number of cells within the tumor that will express the target molecule against which it will actually be efficacy. Now the association with Ayurveda, I think I can skip this slide in the interest of time because it's already been said before. One can actually look at the different types of doshas and prakritis and try to come up to fit them in. But we've already heard of several types of biomarkers, so I could skip this slide also. What probably we could do is something that I thought of, because we have these 3 subclasses, molecular subclasses. We can actually profile the patient's prakriti in these subclasses, and that could be a fascinating tool. We can do it either as a bottoms-up approach, wherein we go in with our predefined classification scheme, or what we have been doing earlier is we've been exploring it in other contexts also, wherein we do unbiased top-down stratification using all the biomarkers that are identified with, or which are being identified, I'll say, with the Ayurvedic system of stratification, which may associate a certain, say, prakriti with a set of biomarkers, and then we can actually find out whether any of these biomarkers are also present in our molecular classes. And this type of an association building, I think that will be crucial to identify a very, very specific case study wherein we can step in and take the field ahead. And I hope this will benefit patients at large. So I think I've really raced through my talk. I hope I made some sense, but the take-home message is that there's a lot of work that still remains to be done before personalized medicine can be realized. And it would definitely be very interesting to define such type of overlaps between what is done under the genomics umbrella and to find out if we can actually overlap this with the Ayurgenomics based approaches, and which can finally help a patient. So I thank you all for a very patient hearing. And this is my group, and we have some artists in the group who keep on doing these caricatures that keeps us lively, at least on paper. Thank you. Am I allowed to have questions in the breakout session? Okay. Thank you.
Thank you, ma'am. Ma'am, I'll request Mitali ma'am to please come on stage and
we have the last speaker for the capsule sessions. And we have none other than Dr. Ghanasham Marda, sir, who is an avid academician, researcher, and practitioner. I'll quickly introduce because the CV would go more than 50 pages. We've cracked them. He is an Ayurveda consultant at Spain, France, and Europe since 23 years now, been actively involved in all the clinical researches across Europe. Sir has co-authored the book on Rasayana, The Art of Healing, Healthy Aging by Dr. Pierre Dwarsky, France. He's the co-author for it. And Sir's areas of clinical expertise and research are specifically heart disease, diabetes, pain management, and mental health. It's a proud to have you here, sir. Over to you.
Good afternoon. Thank you. Sorry, being in between your warm cooking and good food, but I have to. So today, my subject is Ayurgenomics, challenges and unmet need in respect with health and wellness. So basically, I come across to genomics when we found a very good study, an article from Dr. Bhushan Patwardhan about Prakriti. And that's what started to interest me a little bit in that. Further, we went ahead with some research papers, some studies, and it was a really interesting area where Ayurveda could contribute. So basically what I'm going to put in front of you, very specific, what is the genomics, where Ayurveda can go, what are our scope, challenges, and unmet needs. So I'll be brief so that there will be no cheat. So basically, what NIH says, genomics is a study of all a person's gene, including interactions between the genes. With each gene has some specific characteristics, so they interact with each other and they produce certain signaling, certain effects, but most important is it is related with environment. So environment, especially diet, lifestyle, and environmental exposures definitely has some influence in genetics or genome. In fact, John Kelly from NIH, MD, he says that an epigenetic changes happens when lifestyle or environmental factors can cause particular gene to turn off or turn on. So environment, lifestyle, and diet, this is our field, Ayurveda field. And if it is influencing the epigenetics, then definitely Ayurveda would contribute in the genomic scenario in Ayurgenomics. Of course, this tool helps you for some diagnostic things. These tools helps you to find out some pre-symptoms, etc. These tools helps you to design certain treatment modalities. These tools help you to find out some risk factors, etc. But more than that, what Ayurveda needs is something a little different approach. So what I've done, I had some compilation of what we can screen through genetics, and then the scope of Ayurveda. So what genetics is doing? First, diagnostic testing. For example, if you have some disease which the cause may be genetic changes, then there are some diagnostic tools. Pre-symptomatic or predictive approach. If your family has some genetic condition, then to find out earlier, to find out that can transmit to the next generation. Then carrier testing, that is important. That's where Ayurveda has a very important role as well. So if your family has a history of some genetic disorder, such as sickle cell anemia, etc., so you have to think before you think of conception to have a next generation. So that is the carrier testing. Then pharmacogenetics, okay, I don't need to talk, some speaker has already spoken with that, but to find out some dosage medicine, etc. Then prenatal testing, like Down syndrome or trisomy diseases. So you have certain abnormalities with the prenatal age. Newborn screening as well, they do newborn screening. And implantation, like nowadays, in vivo, in vitro, IVF, etc. For that, there are certain testings can be done genetically. So these are the main thing which has been screened as of my knowledge in genetics. And then Ayurveda, as everybody spoke about, the Prakriti thing. And on the base of Prakriti, we can decide the behavioral pattern, we can see the possible disorders, we can see the possible treatment discoveries. But there is some limitation. And when we were having this lunch, there was a discussion about that. Many people asking some questions. So basically, being an Ayurvedic practitioner and a researcher, I know that the traits of Prakriti as an order and the traits of Vikruti as a disorder are different. So you cannot imply as it is Prakriti-based genomics to Vikruti. You cannot. Because, for example, let's say Vata, the motility, the instability, in a Prakriti way, it is different. But Vata Vikruti could be more different than just this. So the traits are different. And this is an unmet challenge. If you go to Google, you go to Ayurgenomics or Ayurveda genetics, everywhere you'll find Prakriti, everywhere. But what is not met is this. This is one thing. Now, second thing, all this is done on the basis of Vata Pitta Kapha. What about the Dvandvaja, means two together? What about three together? Now further, imagine I'm Vata dominance. Vata dominance means what? Vata 98, 1 Pitta, 1 Kapha. Vata 97, 2 Pitta, 1 Kapha. Vata 97, 2 Kapha, 1 Pitta. Vata 96, 2 Pitta, 2 Kapha. So many things. So how could just having a base of 3 things will go ahead? So that itself is a challenge which is unmet. And that's where really genetic will help us. I'm sure about that. So variations and vikruti prakriti, order and disorder, needs to be tackled well through genomics. So this is the limitation of prakriti thing. Now, what are the scope? I told you I'm sticking to just health and wellness, not in the disease, but still it is a big subject. I have chosen few. One of the most important thing, the big scope, is cross-kingdom genetics. Because Ayurveda is one of the few sciences which uses organic material or plant material to treat the disorder, which uses organic material, plant material, food to maintain the health, to prevent. So how this plant material is handling animal kingdom, human material. So there is a big scope of research. There is a big thing required, or this is an unmet need, that plants, I don't need to tell you, many of you are experts beside Ayurveda faculty people, this MIRMA from the plant. There are few studies carried out that how plants are dealing the human disorder through genetics. But there are certain things which are still become an unmet challenge, which I will explain in the next slide regarding plants. One more thing in Ayurveda described is prabhava, means you have a particular calculation of a plant and suddenly you have some different effect. And they say it is prabhava. It's like if, let's say, Prime Minister Honorable Modi ji is going on, everybody will go to the window. Modi ji has not invited, but it's the prabhava of Modi ji that everybody will rush. Similarly, this particular plant has a particular effect which is beyond calculation of rasa, means our qualitative analysis. So here, genetics could definitely help us. Why this plant has this problem, for example. Then another thing, hereditary and this genetically transmissions, etc. So in particular, which we were talking before also about this pregnancy and all. So Ayurveda describes certain diseases as a bija dosha or kula dosha, which not just the immediate parents, but forefathers. There is a description. So this I don't need to explain because genetic is explaining that. But there are certain other factors also, which we Ayurvedic people consider outdated. For example, in skin disease, it's written that if you insult a guru, if you insult a Brahman, if you insult a god, you may get skin disease. All our teachers told us that this is, okay, I don't use bad word, but this is outdated. But now we know that different stressors have a big role in skin diseases. So insulting guru, that time, a guilt feeling, a guilt stressor can bring skin disease. It's a guilt consciousness. So does that still exist or not? How to know? Genomics will help us. There are certain other factors also. I'm not going to elaborate that because of the time. So other factors plus this. Then the third thing is preventive aspect. There are certain descriptions in Ayurvedic text where they say if you do this, probably this genetically or hereditary disease may subside. So does that exist? How to know? Because we don't know before the birth of that particular thing. The person come to us and then we know only. So there is one way is this genetic scenario. So there are many other factors in preventive cares in hereditary factor. The third thing, which is pregnancy. Ayurveda thinks pregnancy as a holy process. It's for the better next generation. It's not just for pleasure, but it's for better next generation. So what Ayurveda has described, this is a very important thing because I was telling you before, this carrier testing. In our classical text, there is description of Shukra, means semen, and Artava, Oham, Ashuddhi, 8 types. And they describe if you are going for conception with these things, there will be particular anomalies. So how to know that? So this could be one good genetic study. This could be a very good challenge for us with the help of genetics. Okay, these kind of anomalies, this shows this kind of genetic scenario, and this Shukra Dushti person is showing same kind of genetic scenario. Fine, it's matching or it's not matching, whatever. So this is another area where we can go. Then in Ayurveda, there is description of every month fetal development in a particular way. And what is interesting, every month they prescribe particular plants. Every month they prescribe particular diet. For example, Panchame Buddhi. And I have in my clinical practice found so many patients, means children with motor expression disorders, just because the mother was under stress in 5th and 6th month. Probably she was exposed to certain medication as well. And this is clearly described. So again, because there is a prenatal screening, even there is embryonic screening in genetic done. So can we use that to see what Ayurveda describes the every monthly development and every monthly plants? Does that have that same effect or not? Does that exist or not? So this is another scope in Ayurveda where we can go on. Then baby care and baby growth. This is another issue. Because you are screening prenatal, you are screening newborn babies. And if you screen with Down syndrome, for example, I have quite a few patients with Down syndrome. Even as I was explaining during lunch, I had many type 1 diabetic children from age 2 till 16, not even in India, also for Europe and USA. And what I'm seeing, and this is on the rise after COVID, that is another thing, up on the rise. So what I'm seeing is there is a great need to come up with some Ayurvedic thing. But then again, there will be asking evidence-based. So please, genetics will help us. In this, genetic will definitely help us. So baby care and the growth, because there are certain disorders, like for example, muscular dystrophies, autoimmune diseases, where we can really contribute well if we get a support. And that is a big area where with genetics, we can come up with some evidence base. Then diagnostic and treatment. Well, this is not my subject, but I will just tell you a little bit about that. There are specific tests, diagnostic tools given in Ayurveda. For example, in diabetes, the Prameha, what we call, which is a compatible condition of diabetes, there are 20 types described. And all these 20 types described on the basis of physical examination of you. And what they are saying, that in first 10 types, they are curable. Forget about cure. Can we control by checking the urine and the person, no matter if it's the same type, because it's same kapha type of 10 types. So this is another aspect. Panchakarma effect on buddhi or rasayana. It is said that it keeps you young even if you are aged. Mitochondria, we know, which is contributing in aging. So does rasayana have effect on mitochondria? Now, what are the problems? This is my last slide and I will finish within time. So Ayurveda goes really from objective to subjective, from molecule to as a whole. For example, we don't take plant particular element or trace element. We use plant as a whole. So need a different approach. You can't go just on with one molecule or one trace element. You have to have, in genetics also, you have to have combination thing. If you want to go with real Ayurveda research. Ayurveda has a very well establishment of gut-brain thing. a very nice explanation, and not just that, even food psychology. With smell, the HPA axis get triggered, the biochemical changes are happening, and the gut microbiota is ready to accept or ready to refuse. These kind of things also explain Ayurveda very well. So could we come up with the help of genetics and gut microbiota and Ayurveda concept of Agni, etc. Then plant research, as I was telling you before, that there is certain things described, like sesame oil, for example, it reduces fat in obese, and the same sesame oil give you fat or increase fat in thin person. So can we establish something? Take the sesame oil, miRNA, take obese person, take thin person, could we correlate something? Could we find the same effect? Maybe it's not, it's wrong. We can say, okay, it's wrong. And there are two more things. One is Ojas, a special concept in Ayurveda, Bala, which is what we consider like a vital response or immune response, etc., immunity. And there is a very big area which we need to work, and it is the need of food. And lifestyle, food psychology. There is the last line. Ayurveda describes very deeply about sleep and about exercise and their effect, not just on immunity, but also various aspects. And today we know that sleep is a very important topic. So could we go through genomics and find out some good correlation? Thank you very much.
Thank you so much, Madam. Many good questions have come up. Even I'm now thinking this session should I attend. Although I have been asked to be in the first session of inherited disorder. I will request Sain sir to please come on stage and felicitate Marda sir.
In fact, I'll just add, sir has already given the solution for the session 4 breakout. I must also add, sir is traveling to Europe at 6. In spite of that, he's here. We are holding on to him, so that's why we are running against the time. So now we will just quickly make some announcements. We have tried to break the group into 4 parts. And basically, it's not much of a task. I'll expect the teachers to lead the way. And of course, all the various leads to be there. And Dr. Mitali will go around and inspect how it is going. And there are 4 things that you have to say. Shweta, just correct me if I miss out something. So you have to make 4 slides, write it on a piece of paper. First is, depending on these 4 sessions we have just had, first slide is on what is the problem statement. The second slide is what are the unmet needs or what is the gap. Slide number 4 is what is the, third slide is what needs to be done. And 4th is action plan. There I'll add, action plan has 3 parts. There's short-term, mid-term and long-term deliverables. And hope is that out of these, we will get out some nice ideas and projects. But of course, we will have the blessings of the university and all the experts out here. So over to Mansi.
Thank you, sir. So we'll divide, for breakout session, we'll divide in 4 groups. I will just announce the name along with the number. So you will be so the groups will be 1, 2, 3, 4. Group number 1 will be for inherited genetic disorders of sickle cell anemia or thalassemia, any of the hemoglobinopathies. Group 2 will be for non-communicable diseases like diabetes. Group 3 will be oncology, and group 4 will be health and wellness. So I'll announce the name of the participant, and along with I'll announce 1, 2, 3, or 4. Please be attentive and listen carefully. So Dr. Tarannum Patel is group 1. Dr. Shambhala Bali is group 2. This is itna fine print kiya hai ki I am struggling to read, so please bear with me. Dr. Meenakshi Randive is group 3. Monica Dattatray is group 4. Dr. Manjiri Deshpande is group 1. Professor Swati Kiran Chobe is group 2. Okay, I'll just say group 1, you can come to this side of the hall. Group 2 can come here, you can start moving. And group 3 has to go upstairs in that corner, and group 4 has to be here. As I call your name, you can get up and go to where your group is supposed to be. Ma'am, we'll see if we can provide some. Yes. अब अपने अपने ग्रुप में तो जाइए, हम देते हैं. So,
can I come in?
Yes, ma'am. Yes.
No, I just wanted to say that you can say tentatively, but if a clinician feels that, you know, they are fascinated, would like to have something to do with another group, give that flexibility to the work people.
Okay, ma'am. So as you all must have heard, VC ma'am just said that we are dividing it into group, but if you have an inclination and if you want to pursue research in some other group that has been allotted to you, you are free to do that. But I would just say, don't find your friends and then go to that group. Choose the group based on the topic, not if your friend is there or not. So Dr. Aarti from Aarti Firke, group 2, Dr. Dipali Manore, group 3, Dr. Sudha Singh, group 4, Dr. Pranita Deshmukh, group 1, Dr. Sonia Kare, group 2, Dr. Ravina Dalvi, I think, group 3, Dr. Saili Lakshman Tanpure, group 4, Dr. Sumedha Suresh Sirsagar, group 1, Dr. Rutuja Jadhav, group 2, Komal Gaikwad, group 1, Devashri Khare, group 2, Dr. Vijay Rahul Nagarle, group 3, Devashri KS 4. I don't see anybody moving around. किसी का नाम नहीं आया क्या? सब लोग चले गए क्या? Dr. Abhishek Girish Taksale, group 1, Dr. Vaibhav Jadhav, group 2, Dr. Narendra Mohare, group 3, Dr. Archana Arun Kulkarni, group 4, Aishwarya Eknath Misar, 1, Suhasini Kadam, 2, Deepthi Mandlik, 3, Mayuri Athre, 4, Megha Gaikwad, 1, Swarali Vagule, 2, Pooja Sachin Chavan, 3, Pratiksha Anil Patel, 4, Ankita Kalel, 1, Darak Vijay Kumar, 2, Dr. Uma Bhosle, 2, Dr. Mayavati Nanavre, 1, Dr. Sanika Dhananjay Patrikar, 3, Dr. Bonle, 3, Ashok Bonle, 3, Dr. Anchal Goyal, 2, Dr. Shreyas Birar, 1, Dr. Vaibhav Rathi, 4, Dr. Anjali Patel, 3, Dr. Vikas Shirsagar, 2, Dr. Aku Nani, 1, Dr. Datta Madhavi, 1, Dr. Suparna, 2, Dr. Ashwini Patekar, 2, Dr. Apurva Tekavade, 4, Dr. Pranjali Nandurkar, 1, Dr. Niveta Pawar, 2, Dr. Manjusha Gumare, 3, Dr. Renju Arnath, 4. Dr. Okay. So now I have some faculty members, Alina Gore Madam, Dr. Chitra Netare, Dr. Karle, and Saint. So they will be in. Will be going around.
Just to add AFMC, so Dr. Mukherjee goes to group 2, The pathology residents, I'll suggest they go to the group 3 oncology group. We have 2 pediatric residents, they go to the sickle cell group. And we have 2 medicine residents, one can go to the group 2 and one can go to group 4. Anybody else who's left out? Preventive medicine goes to group 2. Sorry, sir. From? Just continue. So we'll just tell you. Which your name was mentioned, sir? Which college, sir? Pardon? BJ? Your name, sir? BJ, we have 7 names, so the 7 of you? Or more? You're from which subject, sir? So why don't you join the diabetes group, sir? Anybody else? Yes, ma'am. Sorry? Which scholar? Preventive medicine. Group 4, everybody, this group is? Okay, group 4, I have about already good 9 people there. Group 3 is still vacant. Group 3, please go upstairs. Group 4 is here. Group 1, 2, 3.
So one each.
1 is on my left, ground floor. 2 is on the right. 3 is upstairs, and 4 is again upstairs on the right. Pardon, sir? 2. 3 is upstairs on my left. Oncology group is upstairs. I'll request the resource persons for each group and the lead speaker also to guide them. By you're all in group 1? Good. So by my, excuse me, attention please. So by my watch, it is 3:15. So we will give you half an hour. That's good enough to make these 4 slides. And we will. So by 3:45, I'll expect group 1 to be ready at least. Group 1, you all are group 1?
I think they have all changed.
Yes, sir.
Only 4-5. So you can ask Bapat Madam and Saudi Madam.
So group 3 somehow I see only
6 people.
6 people. Yes, please.
Yes, sir. Yes.
You are all from, may I know from which colleges you all are from? Nobody from Navle or BJ in group 3?
So, Gule ma'am, group 3 is oncology. Group 3 is there.
Group 3 is this.
This is group 4. So each group, I will come around and give you a letter pad and pens, so you can make notes. And if you have, anybody has a laptop in each group, so group 1 has a laptop. Anybody has a laptop in group 1?
Has, group 3 has.
Group 2 has a laptop. Group 3 has a laptop. And group 4, and none of you have a laptop? Okay, so we'll see if we can provide you with one.
So those with the laptop, please prepare the slides. It'll be easier for us to, project, and the Vice Chancellor will be able to see.
We should repeat that. By choice, देखो कुछ भी नहीं चाहिए. Pardon, sir?
The mixer is not, so I'll go and get it.
So I'll just repeat the slides. Anybody, you all have got the slides that have to be prepared. Anybody who wants me to repeat it? I'll just repeat till the laptop comes up, prepared on a piece of paper. So slide number 1 is the problem statement. I repeat, slide number 1 is the problem statement. Slide number 2 is unmet requirements or needs. I'll project it, the slides. Line presentation.
ड्यूरेशन कितना है सर इसका एक बार बता दो उनको 20 मिनट्स ड्यूरेशन ऑफ ब्रेकआउट सेशन 30 मिनट्स 20
3:45 till 3:45.
हाँ हो जाएगा सर सर ऐसा मत बोलो अभी चल रहा है जो चल रहा है हाँ क्या करना है बताओ सर क्या प्रॉब्लम स्टेटमेंट ना हाँ उसी में कर रही हूँ सर गैप एनालिसिस हाँ
That needs to be done.
Next बताओ सर what needs to be done
Action plan. Enter. आगे tab. Short term.
Hi, I think we'll go ahead and start. So I'm from group 1 now, and we were discussing about inherited genetic disorders, especially hemoglobinopathies. So we wanted to, so हम लोगों ने बहुत जल्दी टाइप किया है, so there might be some spelling mistakes, some छोटी-छोटी गलतियां होंगी तो आप छोड़ दीजिएगा उसको. Excuses. Same, sir. 10 minutes, sir. So we were thinking about, we were talking about hemoglobinopathies and taking lead from Gambhir sir's talk. We had a very good group with MD pediatric students, residents, and Ayurvedic faculties and students. And so we all came together and we came up with a problem statement. What they, so what, this is our problem statement. So what I'll do is, instead of just keeping, I don't want to keep talking, I will give the dais to Dr. Pranita here. She is an Ayurvedic faculty, and she will introduce herself and she will continue.
Yeah, namaskar. My name is Dr. Pranita Joshi Deshmukh, and I am representing my college, Sumati Brisha Ayurveda Mahavidyalaya, Hadapsar. Our institution is working in sickle cell anemia since 90s, and we have a screened database of more than 6,000 patients in sickle cell sufferer cases. And we are already establishing a center where we are providing some Ayurvedic medicines to those patients. So the problem statement we identified was dealing with sickle cell anemia by preventing crisis episodes, which is very common, which is getting triggered by any kind of activity, and how we can actually control that with integrative care. So that care can come out from a diet part, maybe some medicine part, or some therapeutics from Ayurveda, like seasonal panchakarmas and a particular medicine-rich diet. So the second point which we identified was beta thalassemia measure by maintaining hemoglobin level. Now that was a problem statement, and Dr. Shruti also mentioned about some of the aspect of maintaining the stability of red blood cell line, cell wall. So that can be achieved with some of the Ayurvedic medicine. We don't know how it really works. So if we can just have some of the inputs from the expert there, how that medicine actually works on a molecular level, that will be effectively established in the patient care. Second thing is, yeah, that can be established, and we can study that as a part of the protocol. The second thing which we identified was about, this is changing. Yeah, the unmet needs. Like I mentioned, we want to prevent sickling crisis. Now, when it comes to the sickling crisis, the major problem is the major affecting population is in a tribal area. So the accessibility and affordability of the patient care is a question mark. And we need to provide some solution which will prevent that sickling episode. So maybe we need to find out some solutions in terms of Prakriti-based variations in that, how a Prakriti responds to that sickling episode. So first thing, we need to start with screening of the sickle cell patients by Prakriti groups, or we can identify them by groups of Prakriti, and we can identify how a particular Prakriti patient is reacting in a sickling episode. Maybe that will give us some of the insights, and we can work around in the protocol that how the particular Prakriti-wise treatment protocols and prevention protocols can be established. And the second was to reduce the frequency of blood transfusion and its side effects. Like I mentioned, we have a center established, and that center is already dealing with 6,000 patients. When we administered our Ayurvedic polyherbal drug, we found out that the concentration of hemoglobin was maintained at a particular level for a longer duration. So automatically the blood transfusion frequency was reduced, and patients' quality of life was improved. So maybe some sort of rasayan activity is achieved in that, and that can be explored more in the project. So what needs to be done? We are now focusing on thalassemia and sickle cell anemia most frequently here. Like I mentioned, we can just group them up in the Prakriti-wise manner and identify the susceptibility on various levels, maybe on molecular, genetic expression level, or there the integration can come in the place. And Ayurveda integration with pain management. We observed that the pain is very integral part of sickle cell anemia and avascular necrosis of femur, or even the day-to-day activities are not possible when it comes to the sickling crisis. Exposure to the environment, the extreme environments like cold environments or even exercise, exertion, they are problematic. So there Ayurveda can come in the picture. And nutritious millet-based diet we had identified because the patient they are from tribal area. So we cannot give something which is very expensive or out of reach. So we are trying to identify something which is reachable, affordable, and accessible for them. So millet-based diet is something which can be done. And the action plan is slightly, it looks like a very simple plan, but it will take a lot of organizations to come together. In our group, we were together in terms of Ayurvedic practitioners, Ayurvedic academicians, researchers, AFMC students, Ayurveda Medical College of Aguli faculty and students, Akrudi Ayurveda College students, Kashibai Nole Medical College faculty and students, BJ Medical College, and we are deciding that there should be a brainstorming meeting where a protocol can be written, and that protocol can also explore some of the possibilities of pilot project in already established sickle cell areas. Midterm goal or objective could be coming up with a protocol and submit that to IEC. Then maybe appropriate funding agency can come in the picture. And like I mentioned, we have 2 or 3 lines of treatment modalities in place. Like first is to identify the Prakriti-wise classification of the sickle cell patients. And second, we can actually see how the next generation is getting expressed in sickle cell. So Garbha Samskar like modalities or some dietary changes or seasonal panchakarma can come in the picture. That can be decided in the brainstorming meeting. And also how to maintain the cellular stability for a longer duration. Long term would be implementation of that project and establishing the integrative care center in the tribal area. Thank you so much.
There was one point here that I just want to highlight early on about this maintaining HB level, and actually Dr. Shruti wanted to speak a little bit about exactly what the problem is at the molecular level. So I will ask us to share, and because we have the dignitaries here, we can keep thinking even when we go back home how we can resolve this, and then we can come together.
Good afternoon. In any hemoglobinopathy that we face, Good afternoon. I am Dr. Shruti. I am MD Pediatric second year resident at AFMC Pune. So in any hemoglobinopathy that we come across, the main problem is maintaining the red cell stability. If somehow we can find out something, for example, in thalassemia, when the red cell is not stable and there is frequent breakdown, the body itself tries to compensate and tries to shift it to a fetal hemoglobin instead of an adult hemoglobin. So yes, but that change is not absolute. That is not 100%, and that causes some problem in achieving the homeostasis. So if by some method something can come up, some Rasayan Shastra or something that we can discover that can maintain the red cell stability at molecular level, then maybe we can come up with a definitive diagnosis and definitive management. And that itself will help us in maintaining the hemoglobin level, and that will also help in decreasing the frequency of blood transfusion. Also, right now, the drugs that we use in beta thalassemia for reducing the frequency of transmission come with their own side effects, ranging from minor side effects such as nausea, vomiting, reduced appetite, and sometimes major side effects such as hepatitis or growth retardation. So in Ayurveda, if we can come up with something, some treatment modality that can just decrease the frequency of transfusion, again, it will come down to maintaining the red cell membrane stability. So if we can come up with this, so even if it is a temporary measure, it is maintaining the blood levels, the hemoglobin's level for a duration of 3 months to 6 months. even that will help us to decrease the frequency of transfusions. Thank you.
There is additional point to that. The more a patient is transferred, the more the amount of blood is given to them, the more they are exposed to blood.
Yes, sir.
So in the long run also, I think even I would support the group in this statement. That is the Ayurvedic group comes through research. Any drug, any molecule that will reduce the frequency of blood transfusion. Even that is, of great help to these patients of thalassemia, who suffer a lot more in later years because of the blood they have received than the disease person. So I think that's a very valid point made.
Sir, I would like to add to that point that we have already developed a polyherbal medicine, which is hardly composing of 6 ingredients, and we are using since almost 2 decades now. And we have observed that the longevity of the patients is improved. Like we are having patients in the age group of 40s to 50s, and also we are experiencing the quality of life is improved. The transfusion rates are reduced. Yeah, with fewer transfusions, you can say. Like earlier, they used to have 2 monthly or 3 monthly transfusions. There used to be increased cases of crisis, but now with our medicine, we have reduced that.
Yeah. So if something is happening for 2 decades, it's really a success story. Patients are also there. So that means you basically need to have you publish this.
Yes, ma'am. Actually, we started this project in 90s with our founder, secretary, Dr. Agaguzu's initiative, and Dr. Katesar was also there.
I'm just saying, have you published?
Yeah, it is published, and we also conducted external research with CCRS.
Okay.
And we compared the other product with our
So
product.
maybe we should do some molecular analysis to
that's what
understand what is the mechanistic basis that could actually give a fillip to this for many people across the, in fact, like he was mentioning, it would be really, really good. So now since NCS is also there, the cellular readouts can be checked by that medicine, or in a mouse we can study, and we can just see the readout as to which pathways are getting molecular level one can study that, right?
Yeah.
So that itself would be a really good aspect.
And in the past, we have also explored some collaborations with NCCS. We have submitted one project to SCMR, but unfortunately it couldn't work out from the funding agency themselves. That time there was no task force for sickle cell anemia, and that's why it got postponed. But now we are ready with our product, and we are also exploring how it works in Ayurvedic parameter. And
and since you said that you got 6,000 patients, and at least 50% of them are also reachable, and it might be worthwhile to do quite an extensive Prakriti and their sickling crisis, the history of those, and try to put the perspective of Ayurveda, is that better explainable. In that case, you might actually have a protocol also for further management of sickle patients.
Also, especially the next generation, would be milder if we use some of the medicines in the antiviral. That we are going to explore. Like we call it as Masanumasi Chikitsa. If we are aware that the next generation is about to have a sufferer carrier, patient in the house, can we do something where we can alter the expression of the gene there? And maybe the milder symptoms could be there, or mild presentation of the disease could be there. We don't know. We need to explore on that way.
That should be done in an ethical.
Definitely. Thank you.
RBC stabilizing agents, talking about classifying people and how they manifest.
Yes.
Three major,
dominance of the drug. Yes.
And I had one word here you can think of in the long term about counseling. I'm not saying marriage counseling. So this is something based on, I don't know, the Prakriti, because these people need to be told. So with that, it is something that can be thought of at the long run.
That is the counseling part, but I will stop here, don't mind, because of the water of time. Any other point from the panelists? We go to the next panel.
Yes, thank you so much for giving this opportunity. In fact, this is what Sikasli Anime highlighted in the budget also this year. Our trustee and board member is recently awarded with Padma Shri for his work.
So
Thank
do
you so
we
much.
have a diabetes book ready, or do I call the book three? Where is the presentation? Somebody has to open it. Mayo? In the meantime, can I ask group 3 to come? Group 3? No, because you know the connectivity goes, we lose the connectivity with process. That's why we are asking for the pen drive only.
Correlation आणि unit डॉक्टर मर्दानी सांगितलं काय लिमिटेशन्स तुमच्यानंतर ही प्रकृतीमध्ये काय लिमिटेशन्स
अश्विनी ना अश्विनी ना अश्विनी download it.
अच्छा तुम्ही
It's open now.
ओपन केलं असेल
त्यांना बोलू दे.
Yeah, good afternoon, everyone. Myself, Dr. Sonia Kale, PhD scholar from Pune District Education Association, Sakhodri Ayurveda College. And we are from group 2, and here we are given the topic of diabetes mellitus and Ayurgenomics. Share screen. Okay. So the problem statement. There's a huge burden and prevalence of diabetes, as Dr. Gautam Gambhir sir have addressed, and a large variation of symptoms we can see as polyuria, polyphagia, and according to Ayurveda also some symptoms are similar and can be correlated, and there could be a further scope of study in this also. Then progression and type of complications for different types of patients. And we don't know which normal person will get diabetes as per modern. And in Ayurveda, there is certain set of hetu or the causative factors which can also be correlated in this to find out as per age, body type, diet, if they're prone to diabetes, we can see. Then next comes the BSL for each person. It is different, as in Ayurveda, this is also said as Purusham Viksha. All markers are different for person to person. Then adherence to drugs and dietary patterns and lifestyle, it varies. So accordingly also we need to work upon and to focus the style. Next, the risk factors according to Ayurveda. We have mentioned few of them here: as sedentary lifestyle, daytime sleeping, consumption of curds, and some other diets like new grains are also one of the positive factors mentioned in Ayurveda. So accordingly, we can also focus upon the risk factors. Next come Prameha is pre-diabetic stage according to Ayurveda. In Ayurveda, different types of diabetes are mentioned. We can say the symptoms are similar, somewhat similar. So of them, Kafaja Prameha, which is called, is actually Sadhya according to Ayurveda. So it has got a more cure rate, what we can focus upon in the increasing context of diabetic population in India. Next are the unmet needs or gaps, what we need to focus. Yeah. It is often incidental diagnosis on OPD basis on, mass screening is lacking in this area. So it is often incidental diagnosis, no markers to predict occurrence or target organ damage like different pathies, diabetic retinopathy, nephropathy. So no specific markers are mentioned there. And which phenotypes respond better. Here also we can focus upon the role of Ayurgenomics and which phenotypes are more prone to. So likewise studies can also be considered. Then health literacy is another phenomenon where rural population is lacking a lot and they need to be aware, because day by day as we are evolving in different eras, people are becoming more sedentary and physical work or exercise in clinical terms we can say is lacking. And here we can see the health literacy should be reached to each and every person from the society. And in Ayurveda, there is one recent publication from CCRS, which is called as Swasthya Assessment Scale. So that is also can be considered as a health card for each and every person. And if at all he is on the border or on the verge of getting diabetic or not, so accordingly such Swasthya Assessment Scale can also be considered. Then which Prameha is Madhumeha. As per Ayurveda, one type of diabetes, type 2 diabetes is one type of Prameha mentioned in Ayurveda. So is it Madhumeha can also be considered in this link. And genetic linkages with Ayurvedic Prakritis. As I discussed earlier, different types, phenotypes can be considered for different types of Prakritis, and accordingly, diabetes can be screened. So now next we focus upon what needs to be done. In this, Ayurvedic Prakriti profile can definitely of help, which can correlate with screening parameters according to modern medicine also. Such comparative study may help us to focus better on what kind of parameters and what kind of health as per Ayurveda are in sync or not. We can definitely think upon this. Then population strategy and high risk strategy of screening at all levels of healthcare, both public as well as in private sector. So like in tuberculosis or some other diseases, this should be on a mass scale screening, as this is an increasing threat, we can say. Then corrective genes discovered and to continue research over it is another area for the focus. Next is awareness building. Again, as I said earlier, like Swasthya Assessment Scale can be done, or health index of the person can be monitored, and awareness can be built as per in terms of heredity or in terms of overweight or obese person who might be prone to diabetes. So such kind of awareness on person to person OPD level can be monitored. Next is customized treatment regimen as per Prakriti or Hetu, which is actually practiced in Ayurveda on day-to-day basis, but it can be focused through Ayurgenomics. It can be more on the evidence-based platform, we can say. Next is lifestyle modification according to each patient. So the short-term measures, what we have just mentioned here right now are the drugs, basically short-term drugs, and dietary and lifestyle changes, because basically as per Ayurveda, diabetes is a lifestyle disease in broad terms, we can say. So along with the drugs, diet and lifestyle need to be modified a lot, and stress management and compliance to treatment are few other points to be considered, and addressing each risk factors and clusters of risk factors should be considered. Awareness building we have already talked about. Then in the midterm goals, what we can focus is intermittent fasting or only 2 meals. This could be an area of research, we could say, because as per Ayurveda, it is told that meals, 2 meals are enough for a healthy person. So if he is or she is a pre-diabetic or a diabetic, then such kind of different types of dietary patterns can be worked upon, or which could be best suitable for the person. Next is prevention of complications. Here also the role is tremendous for area of work, we can see. Then adherence to treatment and regular follow-up is again a challenging question, maybe in the rural sector of the practice. And annual screening for complications. This can be done through different government schemes. For annual screening, people would come if at all they are given some sort of opportunities to come up for different screening assessments, we can say. Then self-care in diabetes, here the food care is of utmost importance as per modern science. And likewise in Ayurveda also different parameters are given, which can again be considered together. And diabetes card to each patient, as it is mentioned, like I sometime before I mentioned the Swasthya assessment scale. So likewise, such diabetes card to people who have got some sort of family history or some sort of elements which are consumed more often, because of which they can be more prone to diabetes. So such cards can be of better use for such kind of patients. And prevention of depression and related mental health disorders is again an area which has got a major or big role, and that can also be focused better with the help of Ayurgenomics, we can say. And the last slide of our presentation is a long-term focus plan or action plan. Here, surveillance and awareness. So surveillance should be on a mass scale. For mass screening, it is quite of help. Then adequate government participation through different national programs like control of diabetes. And in this, different elements can be added as per the combination of Ayurgenomics. Different screening tests can be added, few medicines which can be acted upon, considered as broad spectrum medicines. So such things can be considered. And availability of anti-diabetic drugs, quality of such drugs at all levels, and the duration of such drugs, combining different pathy treatments, or some models can be formed from some modern science and some Ayurvedic factors. Then HLA profiles, mass screening of Prakriti, then reduction to exposure of all risk factors, then national food policies, availability and accessibility of healthy and nutritious food, marriage counseling and genetic susceptibility, as it is a Kulaja Vyadhi as per Ayurveda, and training of the medical and paramedical manpower for the treatment. So, extremely sorry, sir. It is.
One or two actions that has to be suggested by you. This is all the theory that you know what are the gaps and what are the areas where we have to work. That was necessary. It's okay.
Yeah, this is volatile. So I will also recommend extent that is like people continue. It's try to practice mindfulness. So when you take the mindfulness, you have a central theme, and then you will find that you are being repetitive in all your slides. So it will reduce the redundancy and get to the three, four focus points. Because your focus major was prevention. Prevention requires screening, awareness. So these are what use of that. So if you make it in that, then your deliverable becomes fixed and then defined as to what is to be done.
Okay.
So the problem statement is that you need to stratify the patient for risk, for prevention, and for complication. So it could be just toughness in
Okay.
practice like that.
Thank you so much, ma'am. We'll definitely rectify next time. Thank you so much.
Okay. So I'll just add to this what like our panelists have just said. I request that the team leader and the panelists for resource person for group 2 just go back to the things and try to identify like very simple look at the preventive fast, look at the risk assessment, and from my mind, and well integration. So it's very simple, like we have three medical colleges, like 4 Ayurvedic colleges. And just a thought that if there was a AI and machine learning part which Madam mentioned, and she had the software, why can't we get all the OPD or the patients and their data? You have all the data with you. Put it and then see how many have gone into, complications, which are the end organs. It will separate out itself on its own. And then of course you have the various, countries. So to begin with, it's a short term. So thought and like I said, that we have to look at the larger ways that is long term into community based program. So we go into the community. Okay, are we ready?
Yeah.
Ayurgenomics
and cancer. It's okay.
Second, there is a no, cursor.
Okay, so we are group 3 presenting Ayurgenomics and oncology, that's cancer related problems. So unanimously we were a group of 10 people, like from AFMC and various Ayurveda doctors. So unanimously we decided to discuss on breast cancer and ovarian cancer in women, because it's predominantly that is the challenging problem within the woman cancer patient. So the thing is, how can we correlate the clinical subtypes of breast cancer and ovarian cancer and Prakriti and Vikriti? So for the Prakriti, I think, ma'am, you can talk about.
Yes. Prakriti, we in this project, we can have some other objectives also besides this. The first one regarding this is to see the susceptibility of different types of Prakriti towards the particular type of cancers, subtypes of cancers. This is the main objective. Then Prakriti and of course Vikriti. Then secondly, we can see if the patient is under treatment, then which type of complications he is having with the treatment, and can we see the types of complications, and can we correlation with the Prakriti types. Then according to Prakriti, can we suggest him the diet, personalized diet to each and every patient. If at all patient with Vata Prakriti or patient with a Pitta Prakriti, and he is having complications definitely with the treatment, and his predominance of dosha. So can we balance his dosha with the help of diet, that is lifestyle modification, and these things we can see. Then we can counsel him or her as per Prakriti for sleep and each other things also. Then one more thing, as we are seeing the Dehika Prakriti, Vata Pitta, then even we have to check for Manasa Prakriti also, and that tool is still not available. That valid tool is still not available, but that can be prepared, and even Manasa Prakriti can be seen. The sattva of that patient can be seen, and he can be counseled with that, because the stress level will be different in different Dehik Prakriti and different Manasa Prakriti also. So stress we have to think about, which will be the major problem in these patients. So stress as per Prakriti, and we can counsel with the Sattvavajay Chikitsa or meditation with yoga, meditation especially, those patients who can perform this. So this is about Prakriti we can see in these patients. So now I will hand over the mic to Dr. Next, ma'am.
Yeah. So mentioning about this, like Prakriti, in the morning we mentioned about the Kapha, Pitta and Vata, which is connected to kinetic, structural and metabolic. So how this is actually we are bridging the gap. We are connecting, trying to connect with the Ayurveda and the genomics. So how this kinetic, structural and your metabolic activity can be connected through Kapha, Pitta, and Vata. So now, as ma'am mentioned that what is Prakriti, and as we discussed about it, that if there is a predominance of some Vata or Pitta, then it leads towards the tumor. So that we call it as a Vikruti. So that's what ma'am will mention about the Vikruti type.
So when we think about the Vikruti, very Leshokta is described in Ayurveda regarding Arbuda Vyadhi. And we have to describe it when we read in between the lines. And the first motive is to develop the database with Ayurveda regarding Arbuda and cancer. Then the Arbuda Vyadhi is Sannipathic Vyadhi. So all the three doshas are taking part in pathogenesis of the disease. And the sthira bhava of this disease, that is the longevity of the disease, is because of kapha. And pitta is related with the multiple division of the cells taking place in this disease. So we have to think of all these things with this disease. Again, this Vyadhi is a chronic disease, and we have to think of the vikara vigatakara bhava taking place in the pathogenesis of this disease. What Dr. Marda have told, that we have to think of all these things when we think of the genomics while we are taking the research projects. So we have also think of the vikara vigatakara bhavas and the stages which are described in the Sanhitas, the Dhatugata Avastha and all these things, and we have to think of the correlation of the Dhatugata Avasthas and histopathological assessments, and again we have to compare all these with the ERPR positive and triple negative cases and all these things. So we have to correlate on the basis of all these things. So we have planned to do the research with 4 stages. So first will be the animal model with cell line study. Then we have not incorporated all these things in the slides. The second will be the clinical study, which will be a descriptive study to develop the association with what is in the modern and what is in the Ayurveda. So we have to establish the association, and then we have to do the clinical interventional study, which will lead to establish the treatment module in the 4th stage. So we have to do the collaborative research with the 3 branches mainly, that is genomics, Ayurveda, modern pathologists and oncologists. Madam will add something.
Yeah. I would like to mention over here that if you have seen, most of the chemotherapies are derived from the plants, like or the flowers which are there, like for example the wind crystal which is there, which has come from the periwinkle flowers, and for docetaxel, which has come recently, which has been very well established in the head and neck cancers, I can say that it has come from the Pacific yew, which is another plant. So I think plant DNA and the genome, human genome, and the Ayurveda, it has to be, it should have a hand in gloves to come ahead with the proper treatment on Ayurveda. As you can see in the ERPR positive patients, we have the trastuzumab, which has been a Herceptin receptor positive patient, and the patient has to take it for a longer time. But if you come with some special solution on the Ayurveda, which is less cost effective, that is the main thing that we'll have to address, because most of the like for ovarian cancer, where BRCA1 is positive or BRCA2 is positive, we go with the olaparib treatment, and the PARP inhibitors, weekly they have to pay nearly 1 lakh for the treatment. So if in Ayurveda, if you come down with some strong cost-effective solutions with a long-term facility, then I think that will be a really boon for the patients for that matter. And but yes, we need to have a data. So for short-term analysis, we can think about to establish associations between Ayurgenomics and the subtypes of breast cancer or ovarian cancer for that matter. In midterm, we can have some data analysis with the patient's history and how the patients have been treated with the allopathic treatment, or for that matter, ERPF positive and the ovarian BRCA2 positive or the PARP inhibitor treatment. So we need to have that association. And at the same time, because many a times in chemotherapy, it's not only the chemotherapy is given nowadays in the cancer patient, but it is along with the chemo, some targeted therapies are also given. So can we have a combination of these chemotherapy plus Ayurveda treatment? If there are some, then we can have a very good analysis on that. And for the long term, if we get good results, then can we just shift over to the Ayurveda medicines only? So there can we see a paradigm shift from the chemotherapy to Ayurveda treatment, because that is our goal for the in this Ayurgenome. That's what I feel in case of cancer treatment. Thank you.
Thank you. It's quite extensive. I think what you were mentioning and there's a terminology for that. The first thing is to have a common ontological distinction. So the ontology in Ayurveda and ontology in modern medicine are very different, and the way you look at the cancer and the way modern doctors look at cancer might be very different. So the first thing I think we should start with developing a common vocabulary which can bridge these ontologies. Because for cancer, there's a huge amount of TCGA actors, already lots of information are available. So one can really look at the available information as if you have generated in the lab. So that amount of data is there. With clinical and all, and Dr. Chandila is really an expert in that. So you can start to get a feel for whether these bridging with vocabularies can now be connected to TCGA. The moment you get that, lots of reagents or lots of already established things can be repurposed, so it will be a most cost-effective way to reach that. And the connectivity map also has millions of perturbations for large number of molecules, and which those molecules could be mapping to Ayurveda medicine. There is already a database developed by a person called Abhijit Samal in the Institute of Math Science in Chennai. It's called IMPACT. What he has done is he's taken all the Ayurveda literature and looked at the plants, looked at the molecules, looked at the diseases, and tried to make predictions. And huge number of people from all over the world are actually using this database. So then you can start to think that so and so plants are used for cancer, whether they are shared molecules, whether those molecules are there in fact. So there's a huge amount of resource. We just need to kind of
we need to collect. So we need to understand first the mechanism, because right till now only it is all clinical database. The treatment is given. We are now actually getting closer to the genomics and trying to understand the mechanism of each plant structure, how it can be used in the future.
And also, madam, we have to think of the basic principles of Vikruti Vigyan, and we have to correlate that principles also.
That you can do in retrospective cohorts.
Yeah.
But building the vocabulary, because in Ayurveda, when there is a cancer patient, he will not be so interested in first publicity than the it will be a quality of life management. So that would be the more primary focus, and definitely will become again.
So I request all the.
I have one question over here, that there is a conception that the Ayurveda medicines have a lot of heavy metals, and because of that, the patient gets kidney affected, and
Yes.
But how to utilize the drug and how to standardize it, that is a very because the metals are not only the treatment part of Ayurveda, but there are some medicine, only plant-based medicines are there. So that can be utilized for cancer treatment. And then I think no metals can be needed for the treatment of cancer or any part of it. But to get the result faster, metals are being utilized. But that are in the form of nanoparticles.
Nanoparticles.
And those are also time tested, and we have the standard.
New topic for us to collaborate.
Yes.
So it will be great to associate with Dr. Sharmila and Dr. Man for this cancer project and these doctors over here. So
whatever has transpired, and request we should put these notes on whatever points have come in, because we would request and require these, should I call white papers, which will be putting up to the white. Group 4. May I have the group?
Thank you so much.
Thank you.
I'll just introduce the group 4 to you. Their topic was health and wellness through preventive aspects of Ayurveda. They have majorly focused on one single aspect, that is gut microbiome, and various aspects of Ayurveda, that is Vyayama, Divaswap meaning amount of sleep in the daytime, and Ahar aspects of it, Takrapan, and Basti. So various parameters and their effect on the gut microbiome is their basic central idea. Theirs was a pure homogeneous Ayurveda group. So at the end of discussion, it would be great if the pure sciences people can guide them how they can go about identifying the modern parameters for it. I invite the group.
Good afternoon, or well, it is a good evening. Myself, Dr. Divyashri KS from Group 4. Our topic, as told by the madam, it's wellness through Ayurveda. So problem statement, we have selected gut microbiota only because Ayurveda has its main aim is to preventive aspect, swasthasya swasthya rakshanam and aturasya vikara prashanam. So both preventive aspect and treatment part is told in Ayurveda, but we have focused only on gut microbiota. Validation for preventive aspect of Ayurveda through assessment of correlation with gut microbiota, we have chosen as a problem statement. Unmet needs and gap. The impact on gut microbiota, various factors like effect of food, like plant and animal based food, effect of vyayama, that is exercise, both good or bad, like excess of exercise is also bad thing, which is told in Ayurveda in both healthy and ill person. And behavioral pattern like Divaswapna. Divaswapna means sleeping during daytime. Sleep is also thoroughly explained in Ayurveda with both its effects and if what effects it will have on a person if excess sleep or like daytime sleep is done. Then psychological factors like stress and its impact on gut microbiota. These are all the knowledge gaps which we can do research on. What needs to be done? First of all, we need to do a systematic review on the topic of Ayurveda and gut microbiota. We need to search out and find out the gap, like what are the researchers done and what needs to be done, and to establish the relationship of Ayurvedic concepts and literature with gut microbiota and doing proper research on microbiota, and to follow Ahar Vidhi Vidhan. This is a specific concept told in Ayurveda, that is 8 principles of food consumption. It's like Ashtavida Ahar Visheshatan or Ashta Vidhi Vidhan, Ahar Vidhi Vidhan, like Ushnam Ashniyas, Snigdham Ashniyas, and their impact, like why we need to take only hot food or with unctuous food, and what is their impact on the gut microbiota. Third is Viruddha Ahara, the concept of incompatible foods, which is properly explained in Ayurveda and it needs to be found out. Action plan for short term, effect of Takrapana, but that is buttermilk, and Vicharana Sneha, like adding some amount of ghee in routine food. And midterm, Basti Chikitsa or enema therapy. And in the long term effect, we can find out how to reduce stress through yoga sadhana, like meditation, how it affects gut microbiota. So this is it. We have actually taken only specific point and we have tried to explain it.
Excellent.
Thank you, sir.
Excellent.
Sorry, ma'am.
Excellent.
Oh, thank you, ma'am. Thank you so much. Thank you.
Actually, this is good. And this actually that you can start to focus on how you will do such experiments.
Yes, ma'am.
Right? Because that's what it is, and that is how one can move about studying health and health parameters. That is where
Yes.
Yeah, but how many days?
Yeah, we need to. 3 months.
So you can set up people who have environment.
This kind of work for older people also and also for an disease.
You captured very well.
Thank you, ma'am.
So I just want to add a couple of things. Although we discussed during the breakout session, and I didn't mention about that this rule of the gut, because I'm not a Ayurvedic person. Since I've worked on the microbiome, so I can say a little bit on that, what I discussed during the breakout session. So you people believe in that? We all believe that we are what we eat, right?
Yes.
So everything goes to the gut, and that's why it's very important. It has been people have shown, there are a lot of studies, including my own books, that there is a link between what is there in the gut, that's the gut microbiome, and the different diseases and disorders, including diabetes and the brain behavior and behavior of other organs. People have shown. Some of the results have come out. It will be really interesting to see that even in the healthy individual, different preferences or looking into the frequency and frequency and looking into the effect of different diet, whatever you have mentioned, right, 8 or 9, which you prescribe, what happens? And as rightly pointed out, it's not necessary just to look into before, but you have to also see the longitudinal data. You can monitor over a period of 3 months and see what happens. And then you get an idea of.
Madam wants to speak something.
You can get an idea on.
Yes. Link to aa gayi hai, but my office told me ki it's running late, so join. When phone aayega, tabhi join karna hai.
She's on phone call, I think.
So, so which you have to look into, not just the.
You have sent it just now? You have sent just now? Okay.
Hold the conversation for a
while.
while.
Send. I'll join on that. Okay.
One thing I want to clear that work is the main reason. Any diseases, when the network is reduces, the disease can stay down. So that we are.
Seriously, I just want to add that all the points you have listed are very excellent. You can enlist all the points as sir said, like which affect the data.
Yes, ma'am.
That is how they affect the metabolism. So then you can relate with gut microbiota, and everybody said like pre and post, but you can even have the cohorts who are indulged in these practices for quite a long time. And then first you can just show like whether there are differences in the gut microbiota of these sets of individuals who are practicing, for example, the daily during the daytime sleep for years together, and the people who are not involved in that kind of practice. So you can compare and then you can see like whether there is any difference. So the factors which affect gut and thereby affect metabolism, those factors can be a factor. One thing I want to add here, that when we say that we want to look into the, when I said that we will look into the gut microbiome, it's not looking into what are microbes or what are bacteria are there. That's not enough. And most of the people they look into microbes say that these are the groups of bacteria or the phyla which are high and it reduces. That's not enough because these bacterial groups are there, they live in a community. How is the geography substituting? What are the metabolites which are being produced by this bacteria when you eat certain food or whatever you take, and how they are helping the health of the individual? The metabolites of these, the formed by these microbes, how they change, also something which you should probably also look at.
That is the data.
Yes, yes.
That will be
Just one point to the August audience here about the feasibility. Sorry, being from the university, my first question is funds kitne lagenge?
Just one question. I mean, thank you for the appreciation to the group and the idea that we shared. Processing a gut microbiome single sample
Shweta?
can cost anything between 5. Yes, ma'am.
No,
Yes,
I
ma'am.
just would like to, I mean, I need to log off to join the Ayush ministry meeting, but I'd like to just give a message to everyone attending. Can I be heard by them?
Yes, ma'am.
Okay.
Yes,
so, can I say a few words?
Yes, ma'am.
Okay. So firstly, I would like to compliment all the 4 teams. I mean, I heard all your presentations, and to put together so clear, concise concepts, I mean, gives me a real reassurance that this workshop has been, and your bags are full. You all have shopped very nicely. Every one of you, my observation was so effortlessly, you all spoke about Prakriti, about dosha. I mean, whether well, from the Ayurveda spoke of gut microbiota, genomics. So I think we achieved this thing, and it's a very, very welcome thing. I particularly liked the concept in sickle about, the way you all have approached it. And I would like to request my department that our Blossom and Nasical project, which is being written up, can we incorporate some of these ideas even there? And besides that, have prospective, these people can forward, and we must provide all hand holding to help them take this forward, including, whether it's the complications, all of them were excellent. Whatever project they would like to, once a protocol comes in, we will call them to MUHS and have a brainstorming session. The next, this thing was also excellent, where people really, spoke of diabetes mellitus, and I'm sure you would be a very appropriate person to handhold them, because we've already designed one large RCT. We could do a multicentric, spearheading it from there, providing the funding, and getting teams of allopathy and Ayurveda across the state to join in, so that we get real large data. The third, oncos, this thing was also fantastic, and it's so easily doable now that we are setting up the genetic gene health, and as it takes forward, again, a very, very interesting concept. But even the wellness one, I really loved it, because they really wove in, and what if I loved best was them wanting to do a systematic review. And I think that is the beginning, where if that can be done first, we should help from the university a team who would join in to do the systematic review, and then help them develop a protocol to implement this in large numbers. So we have a sample is large. So all these can be taken up as them, whoever puts up can be the PIs, join as co-PIs, and it becomes a large multi. So I'll pen off here with best wishes and compliments to the organizers, the Pro VC who's been there, the senior faculty members who did a lot of mentoring, and all the delegates who attended. So I'd request also the faculty which attended start thinking differently for the PhD colleges. I think this is gone. Thank you so much and all.
Thank you so much, ma'am. We are glad you started this off, and we will take this off from here with your blessings and guidance and the collaborative teams. Thank you, madam, for being with us, even through the distance. Thanks again. I think we're at the end of the program. I would request Honorable Pro Vice Chancellor to sum up a few things, followed by vote of thanks by Dr. Saurav Sen, sir. The question that I was asking is economical aspect of any gut microbiome sampling is about 6,000 per sample. So how can we go about that? We can discuss that later for your purpose.
Thank you, Shweta. So I think today's workshop was really a good workshop for all the that is faculties, the medical faculties, also Ayurveda, and also the science faculties. I think this will be the best thing that at least we'll start from our first protocol, and earliest we will again arrange the second workshop under the hands of Genie Health. Thank you.
So thank you so much. We have reached the end of the day and a very fruitful day altogether. So I have the honorable and the pleasant task of thanking everyone, beginning with the Honorable Vice Chancellor Jimmy Kanatkar, who just logged off. And I think she was the driving force. I mean it literally, because she was monitoring us almost day-to-day basis. And I'm so happy that she had such nice words. And it will fructify whatever she has stated and whatever the team, as a team, when I mean all of us here. I'm grateful to our Vice Chancellor, Dr. Nikum, for being a guide throughout and today monitoring the whole conduct of this particular program. It has been a wonderful evening for all of us. I'm talking of the academic and the professional part. I thank Dr. Vani, he's not here, but without his help and his consent in one single sentence that he agreed that to be for conducting this particular workshop here, it would have not been a success. That is Director NCCS. I thank all the people who are sitting here, the entire team. In the beginning, before I go into the specific names, because it was without your participation, I specifically mean the students, the teachers, and all the basic scientists from various pathies, various subjects. The hope is that we will integrate and work together, like what Madam said in her presentation also, and also Dr. Papat about the silos. We come out of it. I thank our two guest speakers, Dr. Mithali Mukherjee. A lot of people have taken your phone numbers, I know, ma'am. So you'll have a lot of phone calls and messages to, address to. And Dr. Supriya Bhalerao, despite having an eventful day in recent past, she has managed and she has decided to come here. I specifically thank Janil Jha, our Dean, Maharashtra Postgraduate Institute. He's come all the way from Nasik to join us. And Brigadier Mulgund, he's sitting in the last row, but he's keeping a watchful eye on all of us. So, sir, thank you. Fault finding will come in and we'll improve further. So, I thank Dr. Bapat, Dr. Prasad, Dr. Marda. He's gone to Europe. Another 6 o'clock, he'll be off. And of course, Dr. Gambhir, he's also left. Despite his busy schedule, he was here. Dr. Ajay Pillai, he's looking the most tired, but he said, I'm used to it. But the biggest thank you is to you, sir, from NCCS. The support, he's been like rock solid. And his counterpart is Harshal from Pune. So Wing Commander Harshal More. But not to forget the other names, we have Dr. Shweta, Dr. Mansi, Dr. Karle, Dr. Mayur, Dr. Ahir, and Dr. Avle, who have helped us. And the entire team of MEHS and NCCS who have been throughout there. And 2 days back, we were wondering, kya kar rahe hain? And I'm so happy today we can proudly stand, especially me here, to thank you all. So in the end, I'll say the tea is there. Please come. It's been a tiring day, but fruitful. Come and have a cup of tea in the canteen where we had a lunch. Thank you so much. Sir, you want to say something? I think there's an announcement. There's a mic.
Take this opportunity to let you all know that on the occasion of the National Science Day tomorrow, we will be organizing an open week, which means anybody and everybody to walk in and go visit some of our designated labs, maybe the central and then this place in the reception this is open to everybody of all ages, everybody from whatever background is better to visit. The timings are from 10 a.m. to 5 p.m. with the lunch break from 1. So please drop by and spread the word about the parents and other young students who might be interested. Thanks.
So while you have your tea and you go to bed, at least by tomorrow, please fill up the Google feedback form. That's the last statement. Thank you.
मी काढून घे तुम्ही काढ